and Nanomedicine Lab. She's also in charge of the Brain Stimulation and Neuromodulation Lab in the Department of Physiology, All India Institute of Medical Sciences, New Delhi. Professor Jain is currently the Vice President of the Indian Academy of Neurosciences, Delhi NCR chapter. She is also the recipient of the AIMS Research Excellence Award. Professor Jain's research has focused on understanding the effect of prenatal environment enrichment on the functional development of various areas of the brain. She will be focusing on the effect of environment on neuronal plasticity and development of cognitive behavior in her presentation titled Prenatal Music Stimulation Effect on Development of Cognitive Behavior. Good morning and welcome, Professor Suman Jain. We are very glad to have you here. The floor is all yours.
Thank you, Dr. Imran. And thank you once again, Dr. Manjula, for making me a part of this symposium. And thanks to all the delegates, all the participants for making it a great success. I think yesterday, Dr. Manjula was telling the participants were around 230. So that's a huge number. And thanks to all of you. So I'll share my presentation now. Okay. Okay, so today I'll be speaking about a work which a PhD student and an MSc student they had done a few years back, and which is very close to my heart also, and very interesting work, which is prenatal music stimulation effect on development of cognitive behavior. Before I start what we have done, I'll just try to define few terms. What is learning? So learning is the process of acquiring knowledge about the world, about the environment, to which you are exposed to. And as we learn, and therefore with repetition of these procedures which we are learning, due to experience, it is a change in our behavior and in our mental state. And this, because this involves a lot of neural substrate and neural circuitry, so the learning actually the permanent, it happens in a permanent fashion, and therefore changes our behavior. Whereas memory is more that what knowledge we have acquired is encoded, stored, and later retrieved. What we have been working is on perceptual learning. So I'll just define perception, as we all know, is the interpretation of the sensory information. So whatever sensory information we are getting through various senses, touch, through eyes, through ears, or through taste and smell, different senses which are there. So what we interpret, when we see, what we interpret is basically the perception. And perceptual learning then is an experience dependent enhancement of this ability to be able to interpret that what we see, hear, feel, and taste or smell. So like learning, then obviously perceptual learning, since it's an experience dependent enhancement, so it results in long lasting improvement in performing the tasks which are related to perceptual or to these senses. But this perceptual learning is mostly implicit. So we are not aware that this is happening. We are generally not aware, and therefore, but however, we do learn. Like a child, when a child is born, they learn that this is their mother, this is their father, or they also learn that how the mother speaks. So that is why they are able to recognize their mother voice from other voices. Okay? So those type of learnings which are taking place without our awareness is known as perceptual. That is the part of the perceptual learning. It is not only in child that this is happening, this is also seen in adults, and this can be easily studied by a very simple perceptual task. Now this too, during development, so it has been seen that in humans, the hearing is established in utero by third trimester, and there is arousal of fetal activity whenever some species typical sounds will be made. I'll be talking about details in later slides. But what is these observations, these few observations which came up actually led number of scientists to work on these areas. The 2-day infant old is able to recognize their mother voice. and preferentially respond to them. So even if a group of ladies are standing, then they are able to, based on the voices, on the voice of the mother, they are able to recognize the mother. So even if the two ladies are speaking simultaneously, then also they are able to recognize. However, father's voice, recognition of the father's voice takes some more time. Okay. So there is some sort of development of perceptual learning during prenatal period. In utero, these suggest that during in utero, there is a development of perceptual learning, which may affect the postnatal behavior. So these observations which were reported, we got really intrigued by them, and we thought, let's do a proper scientific study and to find out that what is the scientific basis of such observations and how these so-called prenatal perceptual learning can affect our postnatal behaviors. Right? So before we go on to the experimental details, I just want to show that what are the factors which are governing the neural networks and during what times or during in vitro, how the final structure, morphological or functional development of the brain areas takes place. So very crudely, we can divide the whole in vitro gestation period into prenatal and perinatal period. Prenatal is mostly a lot of it is activity independent. That means it is mostly during this time there is a gross laid down of various structures of the brain areas, which are mostly guided by the molecular markers. However, as the gestation proceeds, then and the structures are being laid down, they start becoming functional, and then they can generate their own, they start generating their own spontaneous activities. Like cochlea, if we are talking of hearing, cochlea develops by say 20 weeks in humans, and then they start generating their own spontaneous activities. Now these spontaneous activities then will be able to guide the further maturation of the different structures. Along with this, since the structures like cochlea has been developed and there are laid down of pathways, also neural pathways, they can also respond to the external environment. So they can respond to sensory evoked activities, as we say, or to the external environmental factors. Okay. So this is briefly, and then these environmental factors again fine tune the neural pathways or the structures or the functions of the various areas of the brain. So to study this in detail, the perceptual learning, we chose chick as an animal model. We chose chick because it's a precautious animal. Precautions means that when the chick is born, like in case of humans, most of the sensory systems are well developed. Okay? And another more important reason for choosing chick was that we can get fertilized eggs from the poultry farms, and then they can be kept in the incubators, and we can then manipulate the external environment very easily. In case of chickens, there are basically two major developmental periods. Like I was saying, pre-hearing period, when there is more laying down of the structure of the auditory system, and therefore there are synaptic connections being formed between hair cells and efferents, or between primary efferents with the medulla, and then the cochlear neurons, they develop their own spontaneous discharges. And then there is another period, which is this is embryonic day till 16, up till embryonic day 16. Then after embryonic day 16 onwards, now the system is ready to respond to the ambient sounds or to the external environment. And there is also emergence of the frequency selectivity. They are able to differentiate different frequencies. Okay, so initially the low frequencies and then moving on to the high frequencies. So the first objective of the study was that how does prenatal auditory stimulation, again we chose auditory system because is in the incubator, as I'll show you, it is very easy to play the cassettes. So you can keep a tape recorder and you can play the cassettes, and therefore the fertilized eggs which were kept in the incubator can get stimulated. So how does prenatal auditory stimulation affect the postnatal perceptual learning of the auditory system itself or of other modalities, which could be like visual system? Okay. And if it is affecting any other system, maybe the visual system, then is there any critical period for this cross-modal plasticity? Now, why we chose visual system was that there were reports that during development, onset of function of various modalities, they proceed sequentially, in the sense that tactile is the first to develop, followed by vestibular, chemical, auditory, and visual. And since we were studying auditory system, we thought again more for practical purposes to study the visual system. And then there were studies lately to say that if you're studying, if you're manipulating one sensory modality, it cannot be, should not be examined in isolation, and may affect the development of other modalities as well. So this was the incubator which we had got designed. So this incubator had a tray wherein we can keep more than 100 eggs at one time. And then they had the speakers also. Through the speakers, the sound was coming inside the incubator, and it was a soundproof in the sense that the door was made soundproof, so that no noise can from outside environment can go inside. So it is when the eggs are being incubated, then they are only hearing the sounds which we are playing, right? So, and the temperature, humidity was maintained. They were tilted four times per, in a 24 hours, as generally, naturally it is being done. And then light and dark cycle was maintained. So the eggs were divided into two groups. One was control group, wherein no stimulus, music, any type of sound stimulation was provided. And then there was species specific stimulated group. The species specific stimulated group means that we had got cassettes from Dr. Likita of maternal hen as well as the hatchling calls. So as you can see in control, the chicks, they have the gestation period of 21 days. So no extra auditory stimulation was provided to these eggs. Whereas in species specific groups, it was at 10-day onwards that maternal call was being played, okay, which had a low frequency. Now this was played at this time because as the cochlea had developed, then they were more responsive to low frequency rather than to the high frequencies. So as the maturation proceeds, now they become responsive to high frequencies also, and then therefore we played hatchling calls from 16-day onwards till 21 days. And these were played 15 minutes per hour for 24 hours at 65 decibels. Now to assess the cross-modal plasticity or critical period for the cross-modal plasticity, then the species-specific call stimulated group was further divided into 3 subgroups. Okay. In one subgroup, the maternal calls were played from embryonic day 10 to 16. Only from 10 to day 16, 10 to embryonic day 16. And so this is the time when auditory system is developing and acquiring the frequencies selectivity. Now visual system has been shown to start acquiring the functional development from 16-day onwards, and therefore we provide auditory stimulation from 17-day till hatching. And then there was one group wherein we played throughout the embryonic period, that means from 10 days to 21 days. So this was, A group was when the auditory system was preferentially developing, functionally developing. In subgroup B, we played we provided auditory system when the visual system is preferentially developing. And in subgroup C, it was throughout from 10 days to 21 days. Okay. So for testing the perceptual learning, we did a simultaneous choice test. So this was a big arena, open field arena, which was 160 centimeter to 180 centimeter. And then there were 2 approach areas, okay, which were delineated in this box. Okay. And the tape recorders were placed on both the sides. So from one side, we played chicken maternal call. And from the other side, we played bob-white maternal call. So for testing the auditory perceptual learning, so to be able to test that the chickens which are born, okay, so all the testings were done after birth. Suppose has day 1, 2, 3, we had done the testings. So to test that whether the chicks are able to recognize their mother's voice, we played one which is species specific of them, that is chicken maternal call, and from the other side we played bob-white maternal call. Okay, so that was for auditory. For visual system, then we taxidermically prepared these models. One was quail model, which was obviously non-specific, and then there was chicken model, okay, chicken hen model. Okay. Now, when we were testing the visual stimulus, visual perception learning, then the sounds from both the sides were only of chicken maternal call. And then the tick was left into the center. And during the period of 5 minutes, we recorded that first that how many ticks are responding actually, and therefore have moving to either of the areas. What is the latency of approach in either areas? How much time they are taking to move into either areas, and then how much time they're spending. Okay. So that time, how much is they're spending was also very important. Now, this is about the auditory results of the auditory perceptual learning. So, as I said, that from one side we played the chicken maternal call, from other side we played the bovine maternal call. And we found that most of the chicks, 95% of them, they responded only to the chicken maternal call. Okay. So I'm just showing you the results of those chicks who had responded to chicken maternal call, because their maximum number of them were there. So now these are the three groups. So one is control, which is one is the control, okay, the first bar. Then dark blue bar is the A group, that is when we played the stimulation from 10 to 16 days. White bar when we played from 17 till the hatching, and then black when it was played for throughout. Okay, and this is the different age groups: post has day 1, post has day 2, post has day 3, right? So what we can see that at post has day 1, there was a significant increase in the number of responders. Okay. However, at pH 2 and 3, it wasn't much different among the groups, but definitely there was an increase in control group at post has day 2. Okay. So unstimulated chicks, after 24 hours, they were not actually responding. They were not very responsive to the sounds, but as they mature, as their system mature, they do become responsive. Whereas the stimulated group, all the three stimulated group, they could respond. They were very
Dr. Suman, your
So Jane,
voice.
yeah, your voice is not coming, ma'am. Could you please check your microphone? Still not coming, ma'am.
No, your voice is still not coming.
Professor Jane, could you please unmute yourself once again? No, the sound is not coming. Could you please rejoin the meeting? You have to leave the meeting and then rejoin quickly. Yes, ma'am.
Am I audible?
Yes, ma'am. Yes, ma'am. You're audible now.
Okay, so okay, so uh, can you hear me now?
Yes, Dr. Suman, you're audible now.
Okay, so I'll proceed. I don't know where I had reached, but uh, so I'll okay. Was this clear? That's what
Yes,
protocol we had
ma'am.
used?
We were discussing about different stimulation. Ma'am, I think the slide, 2 slides after this.
2 slides after this. Okay,
Yes,
this one.
ma'am. We were here, ma'am. We were here.
Okay, okay, okay. So, so I was saying that okay. So when we saw the number of responders, when we looked at number of responders, then we found that as compared to control unstimulated group, there was a significant increase in the stimulated group, the number of ticks who were responding. Now, when we looked at the latency of entry, that how much latency they took to enter into the chicken maternal call area, then we found that in the control group, the latency at 24 hours or post has day 1 was significantly higher, and in comparison to rest of the 3 groups, and latency was significantly lower in the group which has received stimulation from day 10 to day 21, that means throughout, okay, as compared to the other 2 groups. Similar were the results in the total time spent in maternal area, that control spent very less time in the maternal area, whereas the species specific stimulated groups spent much more time in the maternal area, and maximum time was spent by the group which was stimulated throughout from day 10 to day 21. So there was a large number of responders, decreased latency of entry, and increased total time spent in the maternal area of the stimulated groups. Now, when we did the visual perceptual learning, and therefore in this, like I had said, that we had introduced the taxidermically prepared models into the arena. So one was quail model and the other one was chicken maternal model. Then we found, and this result was really, really interesting, that at post has day 1, all the 3 stimulated groups were able to, they were responding, okay, and they entered into the area where the chicken hen model was placed. So they were able to recognize the chicken mother, and they moved towards that area. And the latency of this area, the other interesting result was when we measure the latency of entry, then we found that in control group and in the group wherein we had given stimulation only from 10 to 16 days, when the so-called when we say auditory system is developing, the latency of entry was much higher as compared to the other 2 groups, okay, which was maintained at post DH1, 2 and 3. Similarly, the total time spent in maternal area was significantly higher in the groups B and C. B is the group when we gave stimulation from 17 day till 21 day, when the visual system is developing. And the C group is when we had given from 10 to 21. Okay. So in these 2 groups, the total time spent in the maternal area was significantly higher as compared to the other 2 groups. Okay. Now, then we took out the auditory cortex, and then we studied that how is the neural substrate is getting affected, and then we did synaptophysin and PSD95. So in the auditory cortex, we found this is the orange one is the control one, okay, and the rest ones, the brown, blue, and green are the stimulated groups. So in the stimulated groups, the expression of synaptophysin was significantly higher than in the control group. Similar results were obtained when we did PSD95, that in the stimulated groups, the expression was much more as compared to the control group. Then we took out visual cortex, which is known as visual folds of the chicks, And we also did in this left hemisphere as well as the right hemisphere. Now, again, very, very interesting results. So what you see here, that this is control group is orange. The brown color is group in which we had given stimulation from 10 to 16 days. So these two groups, the expression was lower as compared to the other two groups. So although in all the groups, there was an increase in the levels of the synaptophysin or PSD95. So this is embryonic day 19 we have started from. There was an increase in the groups of B and C, but over the time, but there was a significant difference in between the two sets of groups. So that's what we had seen in behavior also, that in the latency of entry and total time spent, when we were doing visual perceptual area, then in the group of control and A, the latency and total time spent was less. Similarly, the synaptophysin and PSD95 expression was significantly less as compared to the other two groups. So this was done in, we also did try to, did post hoc analysis of male chicks versus female chicks. This is PSD95 expression. So when the comparison was made between the left and right hemispheres, then we found that in right hemisphere, the expression of synaptophysin was much more as compared to the left. And when the expression was checked between the males and females, then we found that there was an enhanced expression in females as compared to the males. Okay. So from this part of the study, we concluded that prenatal species-specific stimulation facilitates auditory as well as visual or cross-modality perceptual learning during development by enhancing the neural substrate. However, the prenatal sensory stimulation preserves the species-specific pattern of structural asymmetry or gender bias. So this is, since in the control animals this is being seen, so this pattern of structural asymmetry and gender bias is maintained. It is that this type of sensory stimulation is only enhancing the neural substrate and therefore facilitating the postnatal behaviors. Okay. Now to understand that what is happening to the neurotransmitters, because as we know, GABA, glutamate or BDNF, these neurotransmitters play a very important role in maturation of the or functional maturation of the different brain areas. So we looked at these neurotransmitters. We did Western blotting for BDNF. So we did BDNF. So what you can see, this is in auditory cortex. So in auditory cortex, again, we observed that in control animals, this was the expression of BDNF. However, in stimulated group, you can find that over the period of time, there was an increase in the BDNF expression, and this increase was statistically significantly higher than the control unstimulated groups. Okay, so in all the three groups, there was a significant increase in BDNF. Whereas in visual cortex, the results were quite similar to what we had seen for synaptic proteins. That is, there was a significant increase in the BDNF in both left and right hemispheres in only two groups, that is B and C. That means in the groups in which the stimulation was given when the visual system was developing, right? And again, there was a asymmetry. So in right hemisphere as compared to the left, there was a significant increase in the BDNF concentration. Similarly, in male versus females had much higher the expression of BDNF as compared to the males. Then to study the neurotransmitters, the auditory cortex or visual cortex was taken out, and then we did LCMSMS. And so this is showing you the glutamate. Okay, so in the auditory cortex, what we found that glutamate, in the glutamate, there was not much increase. This is the orange is the control one. Okay, so there was not much increase in control, as well as in the group which is B. B means, that means the stimulation was given from 17 days till 21 days. Okay, so in the group B, the increase till day 16 wasn't actually much, and when the stimulation started, then there was an increase in the expression of glutamate. Okay. And then in all the three groups, there was a significant increase as compared to the control, unstimulated group. Again, in visual cortex, since we started the stimulation from 17 days, the glutamate, and this is the time, as I said, when the visual system is developing, there was dissociation between the two groups. That means control and A group had a lower expression as compared to the B and C group. So that was in both the left and right hemispheres. And then there was a structural asymmetry where in right the expression of glutamate was much more as compared to the left. Similar results were obtained for GABA. So in auditory cortex, all the three groups had significantly increase in GABA. And in visual cortex, we had increase in groups B and C as compared to control and A groups. And the asymmetry was maintained. Similar results with norepinephrine and serotonin. Okay. So this shows that when we are doing giving, there is a critical period of development, that is in auditory cortex. If you are giving from the time embryonic day 10, when the auditory system is developing, then there is a significant increase in the neural substrate, which actually impacts the perceptual and postnatal perceptual learnings. Okay. Whereas in visual cortex, in the groups where the stimulation, if it is provided from when from when it is developing, that is from the 17th day onwards, then it is beneficial, and then it affects the postnatal perceptual learnings. Now, having talked about the perceptual learning about this, then we thought that if this type of auditory stimulation is affecting the perceptual learning, does it also affects the spatial learning? Okay, so a spatial navigation. So the next objective was to study the effect of this type of prenatal auditory stimulation on spatial learning, and not only the species specific stimulation. Then we thought that is it the species specific stimulation can affect the learning, or any other sound, non-specific sound like sitar music in this case. So we played sitar music cassettes when the chicks were when the eggs were fertilized in the incubator. So we played slow music and then we played fast music. So this was again the experimental three groups: control, species specific, call stimulated, and music stimulated groups. In the species specific, like I said, low frequency calls and then high frequency calls. Akin to that, in music group, we played slow sitar music and then fast sitar music. Okay. And the duration and exposure time was again, although similar in all the three groups. Okay. So for doing this, then for testing the spatial learning, then we got fabricated this T-maze apparatus. This work was done by MSc student, Dr. Hina. I think she's also attending this symposium. So we designed this T-maze apparatus. So it has an isolation box, okay, where the animal is, where the chick is introduced. And then from this, there is this corridor, T corridor, so long limb of the T corridor. So and this is a brooder area where the rest of the chicks or the broods are kept. So which helps or which guide this chick which is in the isolation box to reach to the brooder area. Okay, and then there is a mirror also which we placed, and so the chicks have to move from this long arm to the short arm and then into the brooder area. And we measured that how much time it is spending in the isolation box, because once one single chick is kept into this isolation box, it can be an isolation induced stress also. So they may take some time to move out from this box and start navigating. Then we recorded how much time they're taking in exploring and how much is the total time. So then again, the music was played from embryonic day 10 till hatching. And we had three groups and the animals were tested at post-hatch day 1, 3, and 5 on T-maze. So initially day 1, there was T-maze training wherein we had given 3 trials, and then 24 thereafter, we tested their memory or retention. So this slide shows that effect of training on spatial learning. So as you can see, these 3 are showing the different age groups: control, species specific, and non-species specific, which is music in this case. And as you can see, of the 3 trials, so this is day 1. In day 1, we had given 3 trials. So this is the total time. The chicks in all the groups took less time to navigate the maze. Okay, so be it control group or species specific or music group, in all the three groups, there was a decrease in total time taken for navigation through the maze, right? Now, when we compared that among the different age groups, then we found that there was a significant decrease in the total time in the isolation box latency or exploration or the total time taken to be able to navigate the maze. Okay, whereas in comparison to control, 72 hours and 120 hours, there was not much difference. So facilitation of learning takes place only at post hash day 24. Now, when we compared amongst the groups, species specific and music groups with the control group, then a significant decrease in the total time, exploration time and total time taken was seen in species specific, and both in species specific and music groups at 24 hours or post hash day 1. Okay, so day 1 results during training shows that there is a facilitation of spatial learning at 24 hours, and this learning is facilitated when either the species specific or music stimulation is being provided. Then we did, after 24 hours, when we repeated this test, then we found that control and species specific, they were able to learn. However, in the music group, okay, so music group, there was a significant increase in the time. So this increase was there in total time, isolation box latency, as well as in exploration time, as compared to the last trial of the day 1. So there was a memory deficit. Now to see that this memory deficit, is it due to some stress or what is the reason? So we did plasma corticosterone levels also. And what you can see, this is of different age groups. The 3 bars are representing different age groups. So 24 hours, at 24 hours, what you can see, in both the species specific group and the music stimulated group, there was a significant decrease in the corticosterone levels. Okay. Then we also looked at the hippocampus, that what is happening at the hippocampus. Is there any difference between species specific and music stimulated groups? So this is synaptophysin and synapsin. And so what you can see that in comparison to control, there was a significant increase in the synaptic proteins in both the stimulated groups. Okay. So in both the stimulated groups, there was a significant increase, and there was no significant difference between the two groups. Okay. So from these studies, therefore, we can conclude that there is a critical period during which if you're providing sensory stimulation, be in the form of species specific or non-species specific. So basically, when you're providing a sensory stimulation, so that is why I'm using the word sensory stimulation or any sound stimulation, then it is acting as a sensory stimulus. So it can be species specific, can be non-species specific. So it's a auditory sound stimulation. When it is providing during the critical periods, then it facilitates your behavioral development and also facilitates your synaptic plasticity or enhances the neural substrates. It also modulates the activity dependent expression of BDNF and number of brain neurotransmitters. And also that though it is facilitating the behavioral development, enhancing the neural substrate or the expression of neurotransmitters, however, there is a preservation of species-specific pattern of a structural asymmetry or gender bias. My acknowledgements to Professor Shashi Vadva. This was actually, I started working on this project as after my PhD as a postdoc student. So I had joined Dr. Professor Shashi Vadva as a postdoc student, and then I really got interested. It was a very interesting project, and after I took it, when I became faculty, I continued working on it. Professor Rashmi Mathur from Physiology Department, she was also my senior and mentor in Physiology Department. Dr. Nag helped us a lot in morphological studies. Dr. Falguni Anand, you had heard her yesterday. She was the student of Dr. Shashi Vadva, and we both of us worked together on these projects. Dr. Shabuni Rai is my PhD student who did most of this work, and Dr. Hina Kausar, who has done the work on spatial learning. And also thanks to all my mentors, all the students in the brain stimulation and neurophysiology lab. Thanks to all of you for patient hearing, and I would love to have some questions. Thank you.
Thank you so much, ma'am. It was a wonderful session and we really learned a lot from you. Ma'am, I have few questions. How can we adapt the learning from this to human beings?
Sorry, can we repeat the question?
Ma'am, how the learnings from your research work, how can we how can these be applicable for human beings?
Yeah, so you must have observed that nowadays we say that there is generation gaps. The children who are born, they're much smarter than the children who were born earlier. Okay. And so that is one reason that most of the mothers, earlier the mothers used to stay at home and they used to, I mean, not exposed to so much of social media or going out and interacting with a number of, I mean, do not have a number of people around. So the amount of sensory stimulation which the embryos or the kids are now exposed to in utero is much more as compared to earlier. And therefore, there is a preponement of milestones as well as the behavior is changing. Okay. So there is a lot of perceptual learning which is already there. And therefore, the children or the kids who are born, they're much smarter. They're very fast to learn things. And that is why you must have seen that even 1 year child nowadays is handling mobile, isn't it? And, and within 2 days they learn everything, which sometimes for us, if you have to search something in YouTube or maybe it takes time. So that is the effect which is seen in societies. And that is the basis of this. That since in utero, all the mothers are exposed to the screens, to the TVs and other type of social media. We're going to offices, interacting with so many people. So a lot of sensory stimulation in the form of, although I've just shown the auditory stimulation, but visual stimulation also plays a very important role. That can also affect your behaviors or the perceptual learning. Okay.
Right, ma'am. So, ma'am, is there any critical period for the pregnant women when she should be exposed to prenatal music therapy?
Okay. So in humans, I think Soumya, I don't know whether she'll be talking about it, but she has done a lot of work on auditory system. It is around 20 weeks that I think the cochlea becomes functional and it starts responding to the external environmental stimulus. Okay. So I don't know. I mean, I shouldn't be saying that then they can take the music therapy or they can take precautions, but that is when the human embryos become sensitive to at least the auditory stimulation.
Okay, ma'am. Well, that's really nice. So, ma'am, when like the neonatal intensive units, they have a lot of noise also, and the labor rooms also. So, does that have any detrimental effect on neonates?
Yes, actually, I haven't shown the PhD student of Dr. Shashi Vadva had worked on noise, and we had given a noise. We had played noise onto during the incubation, and we found that actually it has a very detrimental effect, and all the perceptual learning, substrate, everything was markedly reduced, much lower levels than the control unstimulated group. So there was a significant effect of noise also.
Okay, ma'am.
Or very those type of jazz music or disco type of musics can affect actually the developmental during the development, embryo development.
Okay. Ma'am, I think Rachina Kapila ma'am also wants to ask a question. Ma'am, if you can just unmute and ask your question. Rachna ma'am. Or if you want, you can type and I'll ask to ma'am. In case any to ask ma'am a question.
I think Dr. Taruna or somebody is asking, their audio is on. Taruna Arora, I think.
Ma'am, that's a glitch. I could see that happening. Yeah, so that's a glitch. Okay, fine. Thank you so much, ma'am. It was very enriching learning for us also. Thank you so much.
Thank you. Thank you. Thanks a lot.
Thank you, Dr. Man.
Thank you so much, Professor Jain.
Thank you.
We'll take a short break, tea break now. I request all participants to rejoin by 11:10 a.m. For Professor Manjula Suri's presentation. See you at 11:10 a.m. Thank you.
हाँ ईमान बोल रहे हैं हाँ मैं I'll just say hello and I'll stop my video. Oh sorry.
Hi everyone, welcome back to the symposium. I'm sure that everybody is here. So the second present for today is Dr. Manjula Suri, who is a professor at Institute of Home Economics, University of Delhi, and also happens to be the organizing secretary of the symposium. She's a medical doctor with a PhD in physiology. Dr. Suri will be speaking on the use of microdialysis technique in neurosciences research. Professor Suri, we are looking forward to an informative presentation. Thank you.
Thank you, Imran. Thank you so much. Good morning, everyone, and welcome to this symposium. And I hope you are enjoying the different talks given by different speakers. I am just sharing my presentation. Double click. The screen visible. So my talk today is on the use of microdialysis technique in neuroscience research. To start with the definition, basically the micro refers to the extremely small scale, and dialysis, as you know, is the movement of chemicals across a permeable membrane. This technique is used to study the biochemical activity in the extracellular space in a behaving animal. It uses the principle of dialysis, which is basically the driving force in dialysis is osmotic pressure. And we introduce
Stop share and share.
sorry.
Share.
Sorry, there is some problem with the screen. No, I should do this. And then share. Is it visible? Is it visible? Okay, thank you. Sorry, everyone. This is there was some screen sharing problem. So I'll start again. I'm going to give a talk on use of microdialysis technique in neurosciences research. Starting with the definition, the micro means it is done at extremely small scale, and dialysis is movement of chemicals across a permeable membrane. And the technique is used to study biochemical activity which occurs in the extracellular space in a behaving animal. And microdialysis basically uses the principle of dialysis in which the driving force is the osmotic pressure. We introduce a membrane into the extracellular space, into the tissues, and then that membrane is perfused with a microdialysate perfusate. This perfusate equilibrates with the extracellular fluid by diffusion along the concentration gradient. And then this perfusate is collected and analyzed by HPLC, MS, or other techniques. First slide, it is.
Participants, just wait for a moment. There is a technical glitch. The slides are not moving. We are looking into it. Just wait for 1 minute. नहीं उससे क्या फर्क पड़ता है?
Right.
ये दो क्यों खुलेगा?
एक तो zoom कर एक बार move करके देखो।
आप एक काम करो, इसको मेरे को मेल कर दो। मेरे को प्रेजेंटेशन मेल करो, मैं उस पर शेयर We have resolved the issue. We are starting in 1 minute. Dekna, slide is moving.
Slide is moving.
Okay.
It is on. Audio is on. Audio is on.
You can start.
Sorry everyone, for there was some technical glitch. The slide was not moving. So I'm sorry, I'm starting again. So I'm going to talk about use of a microdialysis technique in neurosciences research. Starting with the definition, micro refers to the extremely small scale, and dialysis is the movement of chemicals across a permeable membrane. The technique used to study the biochemical activity in the extracellular space in a behaving animal is called microdialysis. And microdialysis uses the principle of dialysis, in which the driving force is osmotic pressure. In this technique, we introduce a membrane into the tissues, and then the membrane is perfused with the microdialysate solution. This perfusate equilibrates with the extracellular fluid by diffusion across a concentration gradient. And then we collect this perfusate and analyze by different techniques, for example, HPLC and MS. This microdialysis probe is implanted into the animals, or nowadays even in humans, stereotaxically. The fluid circulates through the probe, and normally artificial CSF or cerebrospinal fluid is used, which is similar to CSF. And why we use it? Because it causes very little tissue damage. But other fluids are also used, even saline solution is used. Now the soluble molecules, they can cross in either direction, and the molecule cross the membrane down their concentration gradient. There is another technique which is called vitrodialysis, in which the dialysis probe can be used to deliver chemicals or drugs to the brain, and it has clinical application in which the higher concentrations of the chemical are present in the probe, are delivered in the probe, and the molecules cross the membrane and they enter the extracellular space. So a targeted therapy can be given into a specific area or specific space. Now coming to the sampling method, how the microdialysate sample is collected. In this case, the microdialysis probe, it has an inlet tube and an outlet tube, as you can see in the diagram, and it is present in a single shaft. The perfusate flows through the inlet tube to the tip of the probe. The tip of the probe is covered by a semi-permeable membrane, and the molecules can diffuse in and out of that membrane, depending upon the size of the pore of the membrane. Now, if the molecules are larger, like proteins, they are blocked, but the molecule of interest will be moving across the membrane. Now, this perfusate is collected and analyzed by different separation techniques. Now, this is a microdialysis setup, which is, you can see we use in animals. It basically consists of a syringe pump, and this pump is used to perfuse perfusion fluid inside the microdialysis probe, which is inserted into the animal. We have a liquid switch around this tube, which basically regulates the flow of the fluid into the probe. And the microdialysis probe is inserted in a guide cannula, which is implanted earlier. And the dialysate is collected through a fraction collector. And we have a temperature controller to maintain the temperature of the perfusate, which is going through the inlet and collected out through the outlet. Now, before we use microdialysis probe, the calibration of the probe is needed, because the recovery capabilities of the probes, they vary. And this calibration is done in vitro by testing the probe with a known concentration of the molecule of interest in the artificial CSF. So recovery is equal to the outflow concentration upon artificial CSF concentration. In the recovery in brain will not be as efficient as we see recovery in the in vitro experiments. Once we do this calibration, then that probe can be used for collecting microdialysate samples. Before samples are collected, this artificial CSF is run through the probe for approximately 20 to 30 minutes to allow the probe to equilibrate in the tissues where it is inserted. Now, the factors which influence this dialysis rate, these are the surface area of the dialysing membrane, the magnitude of concentration gradient, which is present inside and outside the membrane or the probe. The rate of flow through the probe is very important in maintaining the dialysis rate. Type of dialysing membrane, because different membranes have different pore sizes, and depending upon the chemical of interest, we can use different membranes, and the size of the molecule which we want to check or we want to estimate, that will decide the pore size, and that will influence the dialysis rate. Now, application of this microdialysis technique in neurosciences research is we can estimate the release of neurotransmitters during a behaving animal, and the temporal characteristics of a drug crossing the blood-brain barrier can be checked. Activation of brain areas by various agonist drugs or chemicals can be done by the process of retrodialysis. Activation of one brain area can be done with one probe, and recording of neurotransmitter release from another area can be done with the second probe. So multiple probes can be inserted, and that gives us better understanding of mechanism which is going on simultaneously in two areas of the brain. So, and a single probe can also be used for both delivering or collecting the sample. So dialysis, retrodialysis can be done together in a probe. Now, what are the pitfalls of this microdialysis technique? It's an invasive procedure, so of course it is not free from complications. Complications can occur, like infection. The survival rate is not very high. You need to practice before you are able to perform the experiments. You need to give antibiotic to prevent infection. Then microscopic bleeding can occur. There can be edema, there can be inflammation, and common reaction we see is insertion of microdialysis probe can lead to gliosis. Now, the microdialysis provides the sample, but still it needs another method to analyze that sample. So it's a complicated procedure, and different methods of analysis require different amount of solution. It may be in microliters, it may be in milliliters, and catheter or the probe which monitors the metabolic state of the area is very small. So we are, it's as small as 1.5 cubic centimeter, and in certain experiment, the area of interest may be too small or too big for this probe. So we have to be careful about that. Measurement in this case is not continuous. There is a lag period in between the two readings. And first-hour readings are generally not reliable because we want the tissue and probe to equilibrate, and the acute changes which occur due to the insertion of probe should settle down, and then after 1 hour, the readings are reliable. One thing which is, especially in a country like India, this technique is very expensive. It needs technical expertise, and therefore it is not included in the routine neuromonitoring. Of course, in humans, it is not done, but in animals also, it is a difficult procedure and expensive procedure. Other pitfalls are this microdialysis probe is generally 200 to 400 micrometer in diameter, and the brain implantation leaves a noticeable wound track in the tissue, which I'll be showing you the histological slide where you can see the track of this probe. The implantation generates an immune response, impacting the types and concentration of neurochemicals which are released in extracellular fluid. So when you are estimating these neurochemicals, you have to take into mind that the injury itself, the insertion itself can change these concentration. Now, inserting a microelectrode near a microdialysis probe, the scientists have done that, and they have reported that electrically stimulated dopamine it is eradicated near the probe due to the damage caused by the insertion of the probe. The microglial activation is one of the fastest immune responses to the brain injury, which is caused by this microdialysis probe. But generally, the area of this injury is limited to 300 micrometer in diameter, and microglia are less likely to congregate around the injury. Now, these microdialysis probe can be handmade. They can be, nowadays we have micro fabricated probes. I remember for my experiment, initially we used handmade probes, and by the end of it, we were able to procure micro fabricated probes. They are made from silicon, and they have a porous membrane. They consist of an anodic aluminum oxide layer over an etched polysilicon layer connected to the perfusion channel. Less tissue damage occurs with microfabricated probe as compared to the handmade probes, but the analyte recovery is less than 10% of most of the analytes tested because of the small size of the membrane, small size of the probe. These microfabricated probes are much more reproducible than handmade probes, and they provide an easy route to the customized probe designs. Decreasing the size of the sampling probe is another way of minimizing the tissue damage, which is done nowadays. Coming to this was microdialysis in animals, then coming to microdialysis in humans. The cerebral microdialysis is not used in India or in the developing countries, but in Western countries. It is reported that neuromonitoring modalities are being used in clinical practice, especially in ICU cases, especially in brain disorders, patients of coma. It helps in measuring the endogenous compounds like beta endorphin. We can measure different metabolites like glucose, lactate, and pyruvate. We can measure amino acid. We can measure the markers of cell membrane degradation, for example, glycerol. So the field is really vast. This cerebral microdialysis has a role in the treatment of traumatic brain injury, in subarachnoid hemorrhage, in ischemic stroke, and there are papers published giving the use of this microdialysis in humans. It is also used to investigate the tissue penetration of a drug in variety of tissues in humans, both in healthy volunteers as well as in patients. And FDA supports the use of microdialysis in humans to obtain human tissue drug concentration data. Now, besides brain, the application in other organs, it is used for assessment of bioavailability of topically applied dermatological drugs, and this is even done in countries like India. Monitoring of glucose concentration in diabetic patient by intravascular or subcutaneous probe placement is common abroad. Another application of microdialysis in human is incorporation into artificial pancreas system, which is basically used for automated insulin administration. So this is especially, it has a role in juvenile diabetics who are on insulin, and giving repeated insulin injection is difficult. So this microdialysis along with automated insulin administration is a choice, although it's an expensive technique. Now, application in environmental research, sampling a diversity of compounds from waste, water to soil solution is this microdialysis is used in this. So to check the concentration of saccharides, metal ions, micronutrients, organic acids, low molecular weight nitrogen. Microdialysis has potential even to estimate the fluxes of soil ions, which basically reflect an undisturbed soil environment. So it is used in geology also. Coming to this is the first human cerebral microdialysis was performed way back in 1987 in a patient with Parkinsonism. And this is a picture showing how this dialysis was performed. This is a diagram showing the placement of microdialysis catheter in brain tissue adjacent to a capillary, and how the perfusate is collected or molecule of interest is collected. And it enters the dialysate, which can be a pyruvate or lactate or glutamate, glucose or glycerol, which I have already mentioned. Now, major research area which are involved in microdialysis method is how to collect a sample as rapidly with as little damage as possible. The research is going on to reduce the probe size, to diminish the microglial activation and gliosis, and to reduce the immune response to the insertion of probe. The research is also going on to reduce the flow rates to minimize the damage, to couple it with many separation and detection techniques. So multiple techniques are being used to separate and detect the chemical of interest. To minimize the temporal resolution, to reduce the damage of the implanted probes, which is very common when during insertion the probes are damaged. So you need a sturdy probe. To prevent the damage from the probe insertion, also research is going on to reduce it as far as possible, to decrease the size of the probe, to develop advanced calibration methods, and to segmenting the outflow into droplets, which basically improves the temporal resolution. Now, I'll be now discussing about my research using microdialysis probe. We have used microdialysis technique to study the role of endogenous opioids during the transition period in biphasic response to various pain stimuli in sucrose-fed rat. So the sucrose ingestion leads to a biphasic response, which is generally there is analgesia, reduction in pain during first hour, and hyperalgesia after 6 hours. Dum et al. In 1983, he reported that acute sucrose ingestion for an hour causes increase in release of beta endorphin, and that leads to analgesia. Whereas Mukherjee et al. In our Ames lab in 2001 reported that chronic sucrose ingestion for 6 hours leads to hyperalgesia, increase in pain perception, and it was hypothesized that it may be due to a gradual decrease in endogenous opioid pool. So the role of opioids in maintaining these responses or in the transition pattern from analgesia to hyperalgesia was not clear. Therefore, we performed microdialysis in ventromedial hypothalamus to understand the mechanism involved in analgesia, its transition into eualgesia, and from eualgesia into hyperalgesia in a sucrose-fed rat. Now, the study design for this in vivo microdialysis in VMH during biphasic pain response, for microdialysis, tail flick latency was the test which we have used, and it was noted in 5 sessions at 0 hours, 0.25 hour, 1 hour, 3 hour, and 5 hours in control and sucrose-fed rats at a gap of 1 week. Then the guide cannula was implanted with the help of the stereotaxic apparatus. Again, tail flick was noted in control and sucrose-fed group of rat. Then we gave a gap of 1 week for recovery, and after 1 week, microdialysis probe was inserted in control and sucrose-fed group of rats. Tail flick latency was noted and microdialysis sample was collected at half-hourly interval for 5 hours continuously. This is the setup of microdialysis experiment which we had used. As you can see, there is a ball cage and you can see an animal, the rat, with an inserted microdialysis probe. Now, in this case, the microdialysis perfusate was pumped by a slow infusion peristaltic pump at a flow rate of 1.5 microliter per minute into the microdialysis probe in a freely moving rat. The outlet of the probe was connected to a microtube, and after the collection, it was put under minus 70 degree. The sample was kept at minus 70 degrees centigrade for analysis later on. Now, before this experiment was done, in vitro standardization of the microdialysis probe was done. And this was the diagram is showing the setup used for in vitro standardization. Now, after the microdialysis sample was collected, this is the setup which was used for analysis of the dialysate sample. For quantitative analysis of beta endorphin in microdialysate, ESI MS was used. Basically, it consists of an ionization source, analyzer, and a detector. Microdialysate was introduced into the ionization source by HPLC, and it consists, the HPLC consisted of C18 analytical column, and the mobile phase was acetonitrile in formic acid with TFA plus acetic acid and water. Now, for quantitative analysis of beta endorphin, internal standard was oxytocin, and our external sample was beta endorphin. And preliminary scan of these standards were obtained, and data was used to the data was used by the analyzed by quadratic regression. Then estimation of beta endorphin in microdialysate sample was done. These scans show the time on the x-axis and intensity of ions on the y-axis. And as you can see, no interference was observed at beta endorphin or oxytocin retention window. And the internal standard peak height showed a good correlation with the analyte peak height. Now, these scans show our beta endorphin sample and oxytocin peak of the microdialysate sample. And in our experiment, the limit of detection of beta endorphin was 0.6 microgram per ml. This is the brain showing the site of microdialysis probe, which was confirmed histologically. And this is the histological section of the brain showing canal tip. And as you can see, there is the insertion path injury is visible, a track is visible in the histology. Now, coming to our result, surprisingly, the noxious stimulus given to the rat tail caused an increase in the level of beta endorphin in ventromedial hypothalamus of sucrose-fed rat throughout the period of experiment. This was not expected as compared to the control rat, and it was found to be statistically significant at 30 minutes and at 120 minutes. So to summarize, the noxious stimulus to the rat tail caused an increase in the level of beta endorphin in ventromedial hypothalamus of sucrose-fed rat throughout the period of experiment of 5 hours as compared to control rat, and this rise was statistically significant at half an hour and at 2 hours. Now, coming to the mechanism of this response, which we saw, the sucrose-fed biphasic response, basically it shows analgesia for 1 hour, eualgesia from 1 to 3 hours, and hyperalgesia from 3 to 5 hours. And it is mediated by, as our results suggest, increase in the level of beta endorphin. And it does not support the hypothesis of deficiency of opioids, deficiency of opioids in ventromedial hypothalamus. Now, coming to the proposed model of this response, which we saw, the sucrose induced, it induced an increase in beta endorphin, which may stimulate PAG, which in turn stimulates the epsilon receptors, which are present in the medulla. Now, Nerita et al. Had hypothesized that stimulation of epsilon receptor activates the descending inhibitory influences, which leads to the release of metenkephalin. Now, this metenkephalin stimulates the delta 2 receptors in dorsal spinal cord, and this results in analgesia. Now, whereas hyperalgesia per se may result from activation of the same descending, result from activation of descending facilitatory influences due to the down regulation or desensitization of these delta 2 receptors. And same mechanism may be operating in our sucrose-fed rat, which have shown hyperalgesia at 5 hours. Now, there are a few questions which they arise after our study. So basically, we need to investigate further. One, the role of epsilon, another, the role of delta 2 receptors in this sucrose-induced hyperalgesia. Secondly, we can develop selective receptor agonist or antagonist for sucrose-mediated pain management in chronic pain conditions. And of course, to clinically use initial sucrose-mediated analgesia, which is done nowadays when children are given vaccination, sucrose or anything sweet is given to reduce the pain. Or we can limit or reverse the late hyperalgesia, which is seen in wide variety of pain conditions. So thank you all for listening to me patiently.
Thank you so much, ma'am. That was indeed a very nice presentation. I hope all our participants have enjoyed listening to you and have learned also. Any questions from the participants? Okay, so ma'am, I have come across a term known as DEXA-enhanced microdialysis. So can you explain what is that?
Yeah, this is basically, as I've told you, when we insert a probe into the tissue, it causes injury. So what nowadays, this is a technique in which they give dexamethasone, which reduces the injury to the brain tissue, and we get better results.
Okay, thank you, ma'am. So, ma'am, is the neurotransmitter concentration over or underestimated due to injury due to during microdialysis insertion?
It's both ways. It may increase or it may decrease. So both ways it is possible that we may get a higher value or we may get a lower value. Initially, when there is injury, you get edema and there is the value may be low, but later on it may be high, or both ways it is possible.
Okay, and that can be caused during dialysis. So, ma'am, another question: what is the difference between Zera Netflix method and the conventional method?
There are basically two methods. In one, the conventional method, we just take the dialysate, and in the net zero method, we give different concentration of the chemicals, and then we see we perfuse different concentration of the chemicals, and the perfusate dialysate sample which is collected, we see whether there is gain or there is loss of concentration in the sample. So by that we collect the amount of the chemical of interest in the extracellular fluid.
Okay. Thank you so much, ma'am. That was rather enlightening.
Thank you. Thank you.
Over to you, Imran.
Thank you, Professor Suri. That was very illuminating. Thank you indeed. So the next speaker for today is Professor Soumya Iyengar. Professor Iyengar is a professor and scientist at the National Brain Research Center in Manesar, Haryana. She has authored a number of publications in international peer-reviewed journals. Professor Iyengar's research on the avian black box, the cognitive abilities of the black crow, throws light on sophisticated intellectual skills of the species. Her study of the avian brain provides a new understanding of vertebrate brain evolution and will be the focus of her presentation on the avian black box, the crow brain. Professor Iyengar, we are very, very glad to have you with us today. The floor is all yours, ma'am.
Professor Iyengar, you are co-host now, so you can share your slides.
Hello, am I audible?
Yes, ma'am, you are audible.
Okay, just give me a minute. I'll just share my screen.
Sure, sure, ma'am.
Can you see my screen?
Yes, it's visible, ma'am.
Okay. Now it should be full screen, right? And I'll just move one slide up. So please let me know whether it's visible.
Of course, it's moving, it's moving.
Oh, okay, thank you so much. Okay, so a very good afternoon to all of you. And thank you so much, Dr. Manjula, Dr. Geeta, all members of the organizing committee and Professor Suman Jain for asking me to present this talk at this venue. I have enjoyed listening to both the presenters in the morning, and now I would like to share some of the work that we do in my lab on the avian black box that is on the crow brain. I have put down the names of the students from my lab who have participated in these studies: Pooja, Alok, and Shantmala, all very talented students, and my technicians, Arvind, Krishan, and Ramher, who have also contributed to this work. So why crows? The first time when I started working on crows, it was a very strange question that everybody asked, "Kauwe pe kyun kaam karoge?" Why crows? Well, it turns out that the family Corvidae, which consists of crows, rooks, ravens, magpies, jays, jackdaws, and tree pies, have always been fascinating to me. And this is especially because of their problem-solving abilities. So they are capable of higher cognitive functions also, such as mimicking other species of birds and even counting. And there are numerous examples of what all crows of different kinds, that is the family Corvidae, can actually accomplish. Now, probably some of the earliest studies were actually based on behavioral observations. So all of you would have heard about the thirsty crow from Panchatantra or Aesop's fables. Panchatantra, as far back in India as the 3rd century BC, and Aesop's fables, in fact, more recent, where there was this thirsty crow, which on a very hot day dropped pebbles into a pitcher to raise the level of water so it was able to quench its thirst. Now, this is actually been proven in the lab by scientists Bird and Emery in 2009. You can check their paper, where they have used a modification of the original thirsty crow experiment. In this case, what they used was a long, narrow cylinder and filled it with water. And on top of that was floated a mealworm, which happens to be the favorite food of ravens, which they were using in their study. So now the ravens were not able to get to that mealworm directly, but over time, they learned to use pebbles, drop the pebbles in, raise the levels of water, and help themselves to the food. On interesting variations of the experiment, they were given small versus large pebbles, and the ravens very soon picked up only the large pebbles to be able to get the piece of food faster. And interestingly, when the water was replaced with sawdust and the mealworm was placed on top of it, the ravens did not even attempt the task, showing that they actually understood that properties of different materials are different, and dropping pebbles inside this tube containing sawdust would not have had any effect. So a number of studies, as I said, have been conducted on various on members of different kinds of corvids. One of the examples of their cognitive abilities is object permanence, which I won't be talking too much about today. This is a magpie, which is looking at a toy which has been placed behind a screen. And basically, object permanence just means that the birds know that the toy didn't just disappear or it's not just gone, but it has been placed behind a screen. What is really interesting is that corvids have displayed theory of mind, and that is when an individual can think about another's mental state. This has been shown in a species of corvids, which are called scrub jays. And Western scrub jays are birds which are food storing birds. So for lean times, they actually store their food in different caches all over in the environment. It turns out that amongst these Western scrub jays, it was noticed that some were thieves, those who pilfered others' stores. And what happened was that the Western scrub jays who pilfered other stores were shown in lab experiments that they were more careful about protecting their own caches. They used a number of methods to protect their caches, which are also really interesting. It shows obviously that they are thinking about what they're doing. They avoided spectators while caching. They recached, they pretended to recache and didn't actually cache the material. And they also hid inedible material when they were being observed. Another example of the cognitive abilities of corvids is mental time travel or planning for the future, which was observed in Clark's nutcrackers. Now, these birds store staggering 30,000 caches of food in winter. That is an approximate number. And it has been shown that not only do they remember the location of each cache, but they also remember the nature of the food and first consume perishable food and later the non-perishable ones. Finally, one of the most fascinating abilities of corvids is tool use. This is a species of crows which are called New Caledonian crows. And you can see there is this assortment of small tools that they have made out of twigs. And there is a kind of leaf which is called the pandanus leaf. And they actually carve these tools out of those leaves. So they use these tools to access larvae and other small insects from their environment, which are not normally accessible. And in fact, when a New Caledonian crow called Betty was given a straight wire, given a container, a narrow container with a bucket inside, she was actually able to bend the wire. Obviously, she had never seen this in an environment, but managed to bend the wire, shape it into a hook, and retrieve the bucket which contained the food reward. So I think probably you might be convinced that it's not a good idea to insult anybody by calling them a bird brain. Birds are, at least corvids are, extremely smart among the various kinds of birds. Now let's look at the brain of these birds and see what causes them to be able to perform various functions. Now on the right side is a sagittal section, a half of the human brain. And you can see that there is an outer cortex, striatum and basal ganglia, which is striatum and globus pallidus, which is part of the basal ganglia. Then there is the thalamus, midbrain, hindbrain, and cerebellum at the back. Now, similarly, all kinds of birds, and including crows, have this outer green part, which is called the pallium, which is similar to the cortex, which may constitute as much as 75% of the brain in some species of birds. A striatum, pallidum, thalamus, midbrain, hindbrain, and cerebellum. So all components of the mammalian brain are also present in the bird brain. The one difference is that the cortex, the outer cortex in mammals is 6-layered, but neurons are diffusely organized. They are present all over in the different parts of the bird pallium. And in some areas, they're clustered together to form nuclei. The relative size of the pallium is in fact larger in parrots and corvids. Parrots are another set of very smart birds which have a number of cognitive abilities besides their ability to mimic. And what was also found is that neurons are much more densely packed in the bird cortex as compared to mammals and primates. This is a study which is shown here down below. Now I'm going to point and ask you to attend to just the one in the center, that is the comparison between a raven and a capuchin monkey. The capuchin monkey is definitely larger than the raven, and you can see that the brain weight is also larger. One is 39 grams and versus the raven's 10 grams. But if you look down at these pie charts given below, the green part shows in millions of neurons in the cortex of pallium. And the ravens, not surprisingly, had many more neurons, 1,204 million neurons versus 1,140 million neurons in the capuchin monkey brain. So clearly, birds, and especially those of the corvid species, have enough of the hardware to perform whatever cognitive functions that they need to. Now, this is a really interesting set of circuit diagrams. And this, even though you may not be able to actually read what is given in each box, each of the boxes represents one part of the brain or one compartment of the brain. And the arrows show all the possible connections that you can have in different parts, between different parts. Now, at first glance, they look almost similar. And if you only if you look for a little longer, you can see that the one on the left has more arrows. It is the mammalian brain versus the bird brain. But the basic point of this and the take-home message of this is that birds have all components of the brain which are similar to mammals. They may be slightly different in structure, but the connections are very similar in terms of the major sensory and motor neural circuitry. I come to the part that we have focused on a lot for our studies, and this is what is called the avian caudolateral nidopallium. The caudolateral nidopallium is present at the back or the caudal part of the pallium in different species of birds. And initial experiments showed that when, like for example, in pigeons, they were trained on specific tasks and this area was lesioned, then the pigeons were no longer able to discriminate or make decisions following those lesions. So basically, this area is considered similar to the mammalian prefrontal cortex, which is important for cognition and to perform discrimination, to do various kinds of complex cognitive tasks. Another study which came out in 2005, this is by a very important scientist whose name is Onur Gunterkun. He did these studies initially in the pigeon brain. And what he found was that by injecting dyes into the caudal nidopallium, he found that there were connections with all areas of the brain, like for example, back and forth connections with the visual areas, somatosensory areas, auditory areas, even motor areas, amygdala and other areas. And this is also true of the prefrontal cortex in mammals. It has connections with all other major components of the brain. So this brings me to the topic of discussion today, that is the avian black box and house crows. Corvus splendens, the ones that are commonly found outside, and we can see them everywhere. Now, house crows are ubiquitous in India, and they are actually one of India's best imports, or at least the Indian subcontinent's best import outside, one of the most successful imports. They have gone as far as the Americas. They're present or have been somewhat culled in Australia, but they have populated areas of Africa and even further in the Middle East. So these spread very easily, and they are very, very smart, which is one of the reasons why they have been so successful. Now, when I started my work, there was only one atlas which was available, which was in another species of birds that is called jungle crows. And that was a basic Nissel atlas, which showed the overall parts of the brain. So what we have done is we have taken the house crow brain and cut coronal sections, cross sections. And since we also work on zebra finches, we had the zebra finch brain, songbird brain for comparison. Now, zebra finches are native to Australia. They have, the males produce songs. And these areas that are marked in the upper figure, that is lateral MAN and area X, are two areas. LMAN is in the pallium and area X is in the basal ganglia. These are parts of the circuits which are important for vocalization in adults, some aspects of vocalization in adult males, and song learning in young males. When we did Nissl staining for in house crows, we found these areas were also present. Lateral MN is not present at this level, but it is there in house crows, and you can clearly see also an area X. So obviously house crows have areas for vocalization. And since these areas are important for vocal learning, it is possible that young house crows also learn their vocalizations during juvenile period. Some other areas, like for example, there is this nucleus RA. This is actually important for vocalization itself because it controls the syrinx or the vocal organ. And again, both house crows and zebra finches possess this region in their brains. We next decided to move on to the dopaminergic system. And the reason why we were particularly interested in the dopaminergic system is because it is very important for cognition. To look at or study where this or how this system was spread out over the brain, we used staining immunohistochemistry for tyrosine hydroxylase. Tyrosine hydroxylase is the rate limiting enzyme for catecholamine biosynthesis, and catecholamines, as you know, includes dopamine. Here, what I'm showing you is an example of, this is a picture taken from the substantia nigra and ventral tegmental region in the midbrain of house crows. And you can see clearly that there is high levels of staining here because this area contains dopaminergic neurons. As you know, this is also the case in mammals and in other species of birds. So you can see these neurons here in high power image. Besides the substantia nigra and ventral tegmental area, we have a number of other areas, like for example, GCT area 11. Then there are many cell groups in the diencephalon and in the rhombencephalon. And these, to a very great extent, they of course match the positions of these nuclei in other birds. They also match the same cell groups, dopaminergic cell groups, in mammals as well. Then we moved on to cross sections throughout the brain. And here we are seeing fiber label. This is dopaminergic fibers, which are positive for tyrosine hydroxylase. And really speaking, I should actually say catecholinergic fibers. What you can clearly see is the outer part of these sections are lightly stained, whereas the inner part is very intensely stained. These areas which are very intensely stained are parts of the basal ganglia, which includes the striatum and which basically includes the striatum. These are important not only for movement, but also cognition. And the outer areas, although they're less stained, actually do have fibers. It's just not visible at this low power. And this is the cortical or pallial part of the bird brain. Now, what we were particularly interested in is the caudal nidopedium, because this is considered similar to the prefrontal cortex. And here what we found is that one part of the caudal nidopedium, the medial part, is very heavily labeled with tyrosine hydroxylase positive fibers. And there is a lateral tongue-like area which also stretches out. This is lateral NCL. Both areas have been recorded from in other species of birds like carrion crows, and they are involved in cognition, including making discriminations between auditory and visual stimuli. So other areas that we could also demarcate: dorsal area and intermediate area and a ventral area. So next we decided to quantify our results. What we have done is we have taken high power images of these areas, and we have quantitated the density of the TH positive profiles in each area. The lateral NCL was found to have baskets, very prominent baskets of tyrosine hydroxylase positive fibers around the neurons. Medial NCL, of course, had a very high density of fibers, so significantly higher than other areas. And this dorsal NC was another area which had fairly high levels of TH positive baskets. So we therefore found a way of demarcating between different parts of the caudal nidopedium in our study. So just to summarize what I've told you up until now, the basic organization of the house crow brain is similar to that of other songbirds such as zebra finches, including areas which are homologous to the song control system. Patterns of tyrosine hydroxylase, which is a marker for dopaminergic system in house crows, are similar to those in other songbirds and mammals. And the density of tyrosine hydroxylase fibers can be used to demarcate parts of the caudal nidothelium. Now I move on to another part of the studies that we've conducted in the lab on visual discrimination and self-awareness. So house crows are known for, house crows, and in fact, many other species of crows are known for their abilities at visual discrimination. Something that I've always found interesting is mobbing behavior. This is basically when you go out and injure a crow or injure their young ones. As probably some people even who are listening in might know, the house crow will come back and will attack you specifically out of all the people who are there because it recognizes faces. So crows are known to recognize faces, and unfortunately it has a sharp beak, and even more unfortunately, most of the time it brings its friends and family members along for the attack. And once you are marked down, then it takes a very long time, in fact, not at all, for the crows to forget. In our lab, what we decided to do was in house crows to look for whether they could recognize different patterns besides faces. So what we have done is we have taken two boxes with sliding lids, as you can see in this picture. One has a 6 of spades attached to it, to the lid, and the other one has a 2 of spades. And we wanted to know whether the crow could discriminate between these two. So basically, the crow could slide the lid open and get a reward only out of the 6 of spades. The reward was a piece of chicken. And what we did was we also rubbed chicken inside the 2 of spades box so that olfactory cues were not being used to discriminate between the two boxes. We used to place these boxes randomly across the floor of these cages so that they did not get used to the location either. And I'm going to show you one of the trials. You can see that the bird hops down, goes straight to the 6 of spades, opens the box and gets out its reward. And it achieved a 92% success rate in about 14 days. So quite fast for any animal. Besides this task of pattern discrimination, birds also perform shape discrimination. And we've been studying this recently in my lab. My student Pooja is involved in this experiment. What she has been doing is to take boxes which have hollows carved out in them and placing a food reward in one of the boxes and covering it with two blocks of different shapes. So as you can see here, the triangular box is block is rewarded, whereas the circular shape is not. And this is the piece of chicken which is sitting under it. Now, what we had was birds which were trained to perform to at least 80% of the time they were successful. So this was considered that it was considered that birds were successful at pulling out or learning the task. We also had a group of birds which we studied, which were undertrained. They were only allowed to get up to 50% of the success rate. And we then had controls for this task, birds which were not allowed to form any association with the shapes. They were, for half of the trials, they were given the reward under the triangular block. And for the rest of the 50% of the time, they were given the reward under the spherical shape or the circular shape. And these were randomized. So it was never all given at the same time. Here are trials for birds which are still learning the tasks. So you can see here, the bird hops off the perch, will open the triangular shape first, get the reward, but it still goes over to check under the circular shape. In the next trial, you will see that the shapes have been, or the blocks have been put in different parts of the cage. Again, first it goes to the triangular shape and then to the spherical shape. And in a final trial also, it goes to the triangular shape first and then followed by the circular shape. So what we did after the experiments, that once they performed this experiment, the birds were kept in the dark for some time, that is about half an hour to 45 minutes. And they were then sacrificed and the brains were removed. Now, what we were looking for was an immediate early gene, which is associated with an increase in neural activity. This particular gene that we selected was ARC. or activity regulated cytoskeleton associated protein, which has a number of other functions such as regulating synaptic plasticity, etc., etc. But we were using it mainly because its levels are increased when there is an increase in neural activity. So what we did was we took the brains, cut sections, and then subjected them to immunohistochemistry. Pooja counted the number of neurons in each part of the brain. We knew the boundaries as a result of tyrosine hydroxylase staining in each of these areas, which had been also performed on another series of sections. What we found was that there was a big increase or a significant increase in the number of these ARC positive neurons in the lateral part of NCL, in the stung-shaped lateral part of NCL, in crows which were fully trained compared to undertrained and no association birds. But specifically, the difference was between the lateral NCL and the intermediate NCL. We've also found that there were changes in another part of the nidopallium, and this is the dorsomedial nidopallial area, DMNC. This is this portion of the brain here. And in this case, it was the undertrained birds where there was an increase in the number of these ARC positive neurons versus the other two groups of birds. This is really interesting because it suggests that there might be, while they are learning, dorsomedial part of nidopallium has some role. And then after that, learning may be transferred to the medial or the lateral part of the NCL. But this has to be tested in more series of birds. So to summarize the work that I've shown you up until now, house crows can perform complex cognitive tasks. They can be trained to perform visual discrimination tasks and can also discriminate between simple patterns and more complex visual cues, as well as different shapes. And the lateral and dorsomedial NCL regions appear to be important for learning and problem solving involving visual discrimination in house crows. Now, for the last part of my talk, I'm going to introduce you to mirror self-recognition as a test of self-awareness. It's basically the idea of how one is aware that the image staring back at you from a mirror is yourself and no other. It's not a conspecific that you're looking at. Now here what you have is an example of birds being able to view their reflection in the wild, a group of zebra finches next to a pond of water. And of course, in the urban setting also, you can see crows near panes of glass where they would be able to recognize themselves or at least observe their images. So the way mirror self-recognition or self-awareness is tested in the lab is based on what is called the MARC test, which was developed by a scientist called Gordon Gallup in 1970. Now, what Gordon Gallup did was to test chimpanzees on this task. The chimpanzees had been anesthetized for some other reason. So while they were still under anesthesia, he painted a mark on some part of their head or their chin or just under the neck, where they would never be able to view it by themselves. And the only way, if they started handling this mark or somehow trying to find out what it was, would be in a mirror image. So what he observed was that the initial stages of mirror self-recognition. So once the chimpanzees, of course, woke up, they were provided a mirror to examine their reflections. The initial stages involved exploratory behaviors and social behaviors. So the first, when a chimpanzee has never been exposed to a mirror before, it first thought that the reflection was conspecific and started exploring behind the mirror to see where the conspecific was. It also performed various kinds of social behaviors, thinking that it was seeing another member of its species. This is followed by contingency testing. Contingency testing is basically looking when you first start realizing that perhaps the image is not a conspecific, but it's yourself. What would you do? You would do all kinds of funny actions and see whether the image is also doing that. So turn from side to side, look and see whether the mirror image is also doing it exactly in the same way as you are. And finally, self-directed behaviors using the mirror. If the animal has figured out that this is a reflection, then the animal will start using that not only to touch the mark, but in the case of chimpanzees, for example, they open their mouth and start examining their teeth and picking out bits of food from stuck between their teeth. This mirror self-recognition is, of course, observed in humans and develops in children between 18 and 24 months of age. It is also seen in great apes like bonobos, chimpanzees, and orangutans, but it is not seen in some species such as Old World and New World monkeys, macaques, and capuchin monkeys, unless in some ways they are trained to do so. Interestingly, animals like elephants and dolphins recognize themselves in mirrors, but cats, dogs, and harbor seals don't. And then we come to birds. So for the longest time, it wasn't, people thought that birds just behaved as if the mirror image was that of a conspecific. But a study came out in 2008 where magpies were tested on their ability to recognize themselves in mirrors. A sticker was affixed to the feathers just under their neck. And it turns out that some of the birds, out of a group of 6, 3 or 4 magpies would immediately remove the sticker from under their neck. However, a later study which came out, this was by Solar et al. In 2020, found that these repeated the same experiments and on magpies, and they found that they were not able to replicate the results. They felt that the sticker provided some kind of tactile information or sensation, which the magpies were actually using to pull off the stickers from under their necks. So what we decided to do was to, of course, study house crows, since we had them, but also zebra finches in the lab. And here is the setup that we used. We used 2 cages with mirrors, a smaller one for zebra finches and a larger one for house crows, and a camera to record the observations. So the birds were initially put in there without any kind of spot or dot on them. And also for the control condition, a board was placed over the mirrors so that this was now a non-reflective surface and they would not be able to observe their reflections. We then started putting dots on the heads of the birds or under the neck. So here in this figure, you can see a bit of paint on red paint on the zebra finch's neck or throat. And a dot on the head, and also a black bit of paint on the feathers on the neck as a control. We have also used as a control a dab of just water, which gives it the tactile sensation that paint has been put on the neck, but not actual paint. In the case of the crows, we applied stickers. These are basically bindis, which are yellow in color, and we placed a sticker on the head, as you can see here, on the neck, and here's a black one, which you can almost, it's at least almost invisible to the human eye. Now, what we recorded in these birds was, and of course, not only this condition, before I go into what we looked at behaviorally, we tried in some cases, a board was placed over the mirror, as I had explained earlier. So the experiments, the behavioral results, what we have noted down is the time spent in front of the mirror, the number of songs or vocalizations for zebra finches and house crows, contingency testing, which included head turns, rotation of the body from side to side. Mark-directed preening is especially important. If it starts pecking on the mark to try and remove it, then that suggests that it has recognized the mark. And exploratory or aggressive behaviors, which includes pecking on the floor, mirror, or the mirror frame, and search responses, which is looking behind the mirror and turning clockwise and anti-clockwise. So the general idea when we first started out was that perhaps crows will recognize themselves in the mirror because they're supposed to be smart, whereas zebra finches would not. What we found as a result of our experiments, here's in the case of zebra finches. Zebra finches spent a lot of time, or basically this time spent gives a measure of the attention to the reflection. They spent much more time examining their reflections as compared to a condition where a board was placed over the mirror. And this is also true if a dot was placed somewhere on or the mark was placed somewhere on the feathers. Zebra finches sing a lot, and they obviously vocalized a lot to the reflection as well. And this was significantly higher than in other conditions. They also performed this contingency behavior, that is head turns, much more when they were placed in front of a mirror as compared to the board condition. And finally, they also performed directed preening and pecked on the mark whenever this red dot was placed on the throat as compared to the other conditions. Now, what we found was, however, that it was 2 birds out of our the set of 6 birds that we have tested, which were actually doing this mark directed preening. And here I'm going to show you a video. I hope you can see this. Here's the zebra finch. It's really tiny, so you'll have to focus on this bird. And you can see here, I hope, that the bird is pecking at itself and it is facing the mirror. And this is the kind of behavior that we were on the lookout for. I'm showing you data from the one of the birds which showed more of this mark directed preening. Here is the time spent looking at itself in the mirror versus these are the 2 board conditions in this line graph here. In this graph, what you can see is that there was an increase in this red throat mirror condition. And in fact, the number of preening increased as the trials went on. So this is something that we have found, but as I have told you, this was only observed in 2 of the birds that we studied. Now, what happens to house crows? In the case of house crows, they perform various kinds of exploratory behaviors and vocalization when they are faced with their mirror image. I'm sorry for the loud sound. This is the cooler in the background. But what you can see is that the bird keeps turning around and then coming back to observe itself in the mirror. But when we placed the mark and made it look at itself in the mirror, then house crows were very calm. They just sat there, did not approach the mirror. And although it is grooming itself on its wing, it really doesn't attempt to remove the mark in any sensible way. So none of the birds that we studied removed or even attempted to remove the mark. This is not really part of the experiment, but I thought I would show you this one example that we have of a jungle crow, since we have jungle crows also in India and these are wild caught birds. So along with in our house crow collection came a jungle crow. So we decided to test it for mirror self-recognition as well. I have never really seen a bird performing taekwondo, but you can see here this bird and its very aggressive response to the reflection. So in this video, in fact, by the end of it, we had to actually stop the experiment because otherwise we were in danger of losing the mirror. And you can see that the response of the jungle crow is clearly does not recognize that that is its own image and thinks it's a conspecific male or conspecific bird. So when we quantified the data of the house crows, what we found was that these birds definitely paid much more attention to themselves in the mirror. That it is the time spent in front of the mirror was significantly higher than any of the board conditions. The number of vocalizations that these birds made to their when they were faced with their reflections was also more. And the number of head turns, turning from side to side, this is contingency testing, was more. That is, they did appear to look around to see whether it was themselves or their reflection. But we never found any kind of self-directed preening during the mark test. These findings basically suggest that house crows do not recognize themselves in mirrors. So just to summarize the last part of the talk, basically what I've shown you is that a very small number, 2 out of 6 zebra finches appear to recognize themselves in mirrors and pass the mirror self-recognition or mark test. But 2 out of 6 is not really satisfactory, and this needs to be tested in a much larger sample of birds. Now, it's not only true of zebra finches. Many of the other species of birds and mammals also, it's not all members of the species that pass the mark test. some members appear to be able to do it and others not. And then we have house crows, and despite being able to solve complex cognitive tasks, including pattern discrimination, etc., house crows do not recognize mirror images as their own reflections and appear to treat them as conspecifics. We were feeling a little bad about this, but then it turns out that New Caledonian crows, which are extremely smart and fashion those hooks and tools that they can use for their pulling out grubs, etc., they also do not pass the mark test. They, however, can use mirrors to retrieve food from a hidden location. And this is something that at some later point we might try out in house crows as well. And so finally, the summary of this data is that different levels of self-awareness are present in different species of birds, and also different levels of self-awareness amongst members of the same species. So with that, I'd like to stop there. Thank you all for your attention, and also a big thanks to all my lab members who helped in this project.
Thank you, ma'am. That was really fascinating to see the research you have done and also the kind of cognitive abilities that these corvids have. So yeah, I think we've explored a lot of things in them.
Yes.
Ma'am, I have a few questions for you.
Sure.
So is the avian black box research going to shed light on the origin of cognitive ability?
Well, the cognitive abilities have been studied in various species of crows, as I said, and my studies provided the anatomical details of these parts, especially which were involved in solving cognitive tasks. But I have to say that a very large number of studies is being done where people are recording beautiful studies on electrophysiological recordings. And they have recorded from these areas in other species of crows, like carrion crows. There's a big lab in Germany which is doing this kind of work. And they have also shown that similar areas in the carrion crow brain are important for all kinds of abilities of crows. So it would be really nice to replicate them in house crows as well.
Okay, ma'am, and do you think there has been a convergent evolution of brain and cognition in birds and also in humans?
It could be convergent evolution, and also it could be that the cognitive abilities have evolved independently at different times across evolution. So convergent evolution, of course. And the fascinating part about birds and house crows in particular, or crows in particular, I should say, not just house crows, is the fact that with software which is or hardware which is very different, the organization of the brain being fairly different, no six-layered structure or anything in the cortex, they're still able to perform these kinds of functions and really well at that.
Okay, thank you, ma'am. I think there's Vidya Shukla. She wants to ask you a question. Please unmute and ask, Vidya.
Good afternoon, ma'am. Your presentation was really informative. I myself also work with the zebra finches, and when I saw the results that you presented about crows and the marked recognition test, I was wondering, ma'am, zebra finches being pair bonding species, and as far as I know, crows are not. So
no, crows are also corvids by and large are also pair bonded.
Okay.
Yes.
I was wondering maybe that would be the reason, because in zebra finches we see that they bond for life, and therefore the appearance is more important somehow because of that. So I was thinking if that is something that could play a role in the recognition, mark recognition test. That was something I was wondering.
It is definitely possible, considering that, as you probably know since you work on zebra finches, that zebra finches are sexually dimorphic. The males have a lot of colors in their plumage, whereas females don't. We actually didn't test, we should test at some point mark recognition in females as well. Crows, on the other hand, you cannot tell a male from a female. So perhaps that difference in mark recognition comes from this, but I'm not really sure. And again, the number of zebra finches that actually did the mark directed, task were actually a very small number. So we would definitely, want to test this out in a much larger sample before we can say anything definitive.
Okay, thank you, ma'am. Vidya, I think that fulfills your question. Okay, ma'am, there is another question in the chat box. Does other birds also show a similar discrimination behavior?
Other birds, other species of birds?
Yes, ma'am.
Do they show mark-directed behavior?
Discrimination behavior, yes, ma'am.
Yeah, so as I was explaining, this was first shown in magpies, but again, especially in different species of crows, this is fairly controversial, because one group comes along and says that this has happened, and then another group tests mark recognition and they don't find it so. So even magpies, where for the longest time it was thought that they actually recognize themselves in mirrors, it turns out that probably they were using tactile cues rather than a visual cue to see themselves or recognize themselves in mirrors. So magpies certainly have been tested. New Caledonian crows, as I said, don't recognize themselves despite all their other abilities. They don't recognize themselves in mirrors. So perhaps this is something which is common across different species of crows. There are a number of other species which have also been tested. And I can't remember the name of this one species, where they used, rather than a mirror, a plain mirror, they used a blurry mirror. And it was found, these were the Clark's nutcrackers. It turns out that Clark's nutcrackers recognize a mark which is placed on their body when they are faced with blurry mirrors rather than a plain, clear mirror. Now, why this would be is that, after all, the birds, when they are in the wild, would be observing themselves in pools of water where the reflection is not clear. It's not like a mirror reflection. So perhaps it's a good idea to actually test out blurry mirrors rather than plain mirrors, and then see whether we get the same kinds of findings.
Right. A lot of future scope for research.
Yeah, certainly.
Thank you so much, ma'am. Over to you, Imran.
Thank you very much indeed, Professor Iyengar, for this fascinating and invaluable presentation. Really glad to be present at this occasion. So,
wait, Imran. There is one more question. Ma'am, what is the difference between object recognition test and this setup that you have used here?
The object recognition test, what we are doing is we are placing the birds in a cage, and we have these, as I showed you, two boxes which are randomly rotated in the cage at different positions they're placed. And we put pieces of food under different blocks, and we look for whether they can recognize the rewarded shape versus the non-rewarded shape. With the mirror self-recognition task, we place the birds in a container which has a mirror in it, and we either put a mark or don't put a mark, put a board or don't put a board in to prevent the bird from seeing its reflection. And so one is self-awareness, whether it is aware of itself. The other, of course, is awareness of shapes and patterns in its vicinity. And clearly the birds are able to recognize patterns and shapes and all of that. I also explained about mobbing behavior. After all, being able to recognize faces, or human faces in particular, is no mean task, and which birds definitely do. All species, many species of crows have been reported to do this mobbing behavior.
Then you can take over.
Any other questions?
No, ma'am, that was the last question. Thank you.
Thank you so much. We'll break for lunch, and I request all participants to please rejoin by 1:30 p.m. for Professor Praveer Kumar's presentation. Thank you so much.
So, what is happening? I'm sorry, I don't know what is happening here. Just a moment, please. Okay, so my talk is related to protein misfolding, its aggregation in the neurodegenerative disorders. As most of us knew this fact, that there is a major culprit behind most of the neurodegenerative disorders are protein aggregation or misfolding. It's not moving. So let us start with the physiology behind the Alzheimer's and Parkinson's disease. I'm just going to give you some glimpses because most of you probably aware with the physiology or pathophysiology of the AD and PD. So AD stands for Alzheimer's disease and PD stands for Parkinson's disease. So here you can see that there are several stages of the Alzheimer's disease, starting from the normal to severe Alzheimer's disease. So here, means like, the biggest issue with the Alzheimer's disease is the cognitive declination and the dementia, because, sometimes people are asking that, okay, what is my name? The person is standing in front of home and they're asking, and he or she is asking, do you know where is my home? So this kind of memory decline. Whereas, so this is exclusively a memory deficit disorder, whereas the Parkinson's disease is due to the motor impairment. What does it mean? That you cannot stand properly, you cannot walk properly, and in the later and severe stages, the person is completely wheelchair or bedridden. And the most important thing with this AD and PD, if you can compare AD and PD, in AD it's a memory decline, in PD it's a motor skill decline, motor impairment. So most of the time, these two patients are dying not due to the natural death, rather they are dying due to the accident. So I'm going to start with this background. I'm going to start how misfolded protein or aggregated protein leads to the severity of this disease and how we can reverse. So I start from my lab projects. There are several projects that are going in my laboratory. I'm specifically focusing on the molecular chaperones and ubiquitin E3 ligase pathway. Why because if proteins are misfolded, it has to be cleared. It has to be properly folded. And that is why I'm more focusing on the chaperonic pathway. And if it is not properly folded, it has to be discarded through the ubiquitin proteasome system machinery, UPS machinery. So in the next slide, I'm going to show you what is happening. I don't know why this slide is yes. No, it's okay. So, there are about 70% proteins which are produced inside our cellular milieu is misfolded or not properly folded. And what is happening because of that, it has to go to the molecular chaperones as well as the UPS machinery in order to get a proper folded. So what is happening, this misfolded or unfolded protein cause the protein aggregation that requires a machinery which can maintain or which can give the proper shape of these proteins. And for that, what is happening, you need cellular systems require a machinery called chaperonic machinery or UPS. But the important point here, that which proteins should be discarded from the cellular milieu and which protein should be kept inside the cell is very important. And thus it is only decided by the ubiquitin E3 ligases. And in the next slide, I'm going to show you how these cognates are functional. So this is the UPS machinery. What is happening? E1 is the initiator, E2 is the conjugator, and E3 is the ligator. So what is happening that if a protein is marked that, okay, this protein should be discarded, it is starting the process initiation, then conjugation, And then what is happening? A lysine molecule. Why this is coming in? Lysine molecule are attaching to the targeted protein that has to be degraded and make a polyubiquitin chain. And thus the cell is understanding that, okay, it has to be degraded. But this procedure is not continuous. It has to be stopped one time. And thus deubiquitination enzyme come in action, and this entire ubiquitin molecule, which is recruited by the lysine residue on a targeted protein, is disintegrated and the entire reaction is stopped. So this is the glimpse of the UPS machinery. This is again the same diagram, but it's little advanced, because here I have discussed about the A beta 42 to A beta 4042 and A beta holo-APP. So why these two proteins are important? Because of AD pathology, because they are directly involved in the AD pathology, and you can see there is a many lysine residue, because for the ubiquitination, for the recognition of the ubiquitin E3 ligase, you require lysine residue. And here you can see that in holo-APP there are around 8-9 lysine residues which might have an impact. It is not so far reported. Some of them are reported, like this 351, lysine 351. But in the A beta peptide, which is 40 to 42 A beta peptide, there are two important residues, that is the lysine 16 and lysine 28, which is responsible for the recruiting of ubiquitin on a targeted protein, which has to be discarded. Most importantly, we know that A beta 1 to 42 polypeptide is very, lethal for our brain or our body. Even right now we have the A beta monomer, but A beta monomer is not it's not cause any problem. But the moment it can oligomerize and fibril, the problem is started. Suppose I can give you an analogy that, okay, suppose if you have two synapses are in communication, and if there is a shield between the two synapses, there will be no more neurotransmission, there will be no more communication. What would happen then? Then the synapses will start dying. And this process is called the neurodegeneration or neuronal loss. So A beta 42 is the main culprit for Alzheimer's disease, Parkinson's disease, and inclusion body myositis. Although inclusion body myositis is a muscular disorder, but since A beta is expressing in the muscles, that is why we have taken this also as a consideration as a positive control for our work. You know what we have done? We have started a cellular machinery to understand the A beta etiology and how we can reverse the effect. And for that, what we have taken, we have made some A beta viruses under the control of Teton-Tetoff system. So what is the beauty of the system? The moment you add the doxycycline, the expression of A beta is switched on. So what we were doing over there, that we are using the A beta virus, adenovirus, and A beta Teton virus. So we are co-infecting the cells with these two viruses, and whenever we wanted to have the expression of A beta, we were adding the doxycycline, and whenever we wanted to remove the expression, we are withdrawing the doxycycline, which is because this doxycycline is working on the tetracycline responsive element, TRE element, and that is why you have that liberty to switch on and switch off the A beta expression. And then we have studied many signaling pathways, starting from chaperonic, ubiquitinic, apoptotic, and cell cycle pathway. And here I am showing you what we have done. So we have taken the SHSY5Y. This is the neuroblastoma cell line, and this neuroblastoma cell line is infected with the this CHIP virus. What is CHIP now? Because CHIP is the ubiquitin E3 ligase, as well as CHIP is the molecular chaperones. So CHIP is very important molecule for us because it is triazing the molecular chaperone pathway as well as the ubiquitin proteasome system pathway. So in our system, we believe that CHIP is very important for the reversal of A beta level. If the A beta level is overexpressed, it can be reversed by using the this adenoviral chip. So you can see it here in this panel that 1, 2, 3, 4, 5, 6 in the sixth panel, there is only A beta expression under the influence of doxycycline. If you can concentrate on the lane number 4, you can see that we have everything. We have A beta expression as well as the CHIP expression. And if you compare lane number 4 versus lane number 6, you can see the expression level is drastically dropped down the moment we added the CHIP. Likewise, we have also studied the different heat shock proteins levels, and we have found also under the influence of heat shock protein HSP 70, 60, and 90, A beta expression is strikingly dropped down. And this is the densitometric analysis in the right hand lower panel. So we concluded this that CHIP, HSP 17, HSP 90 attenuating the A beta 1 to 42 level, which is very important for us because this A beta, if we somehow we are able to drop down the A beta expression, we may uplift the neuronal morphology, neuronal physiology. Then we thought that, okay, if this is the case, whether these two cognates are interacting or not, if this level is dropped down, does CHIP interacting or heat shock proteins are interacting with A beta? And for that, we have done the immunoprecipitation experiment. And you can see in all the 3 panels, CHIP A beta, CHIP HSP 70 A beta, and HSP 90 A beta, that yes, under the influence, under the expression of CHIP, we have successfully dropped down the A beta expression. We have lowered down the A beta expression in all the 3 cases. So the take home message of this slide is CHIP, HSP 70 and HSP 90 interacting with the A beta 1 to 42, as well as it is reducing the overexpression of A beta. We also thought, okay, if this is the case, whether it's interacting with the Euclid machinery, and yes, we have proved that this A beta and CHIP, I'm unable to show you here because this, anyway. So here we have also shown that A beta and CHIP are interacting under the influence of 20S proteasome. This paper, okay. Now come to this panel. We have done so far whatever experiment we have shown you in the previous slides are on the neuroblastoma cell line. But in this slide, what we have done, we have used the primary cell culture of the cortical neurons. So what happened, we took the pups, we removed the brain, we removed the hippocampal area, we prepared the neurons from that under the neurobasal media enrichment, and then we have studied the neuron. And here you can see in all the 4 panels, concentrate on the left panel where there is no CHIP. And you can see in the top left panel that under the CHIP, there is no doxycycline, cells are happy. But the moment we have added doxycycline, the cells are under huge stress, and you can see in the black arrow, this is the death, and there is no CHIP. While in the right-hand side, in the lower right panel, you can see when we added the CHIP, you can see the neuronal progressions, the processes are generating. So here we have concluded that indeed, CHIP is very helpful in the primary culture also. Then what we have done, we have scrapped these cells and we have prepared the protein, and we did the expression analysis under the through the Western blot, and we have successfully shown that yes, presence of CHIP rescue the neurons from the A beta 1 to 42 toxicity. This paper we have published in the Human Molecular Genetics and then in the Journal of Neurochemistry in 2012. Now, we thought that, okay, the protein aggregation is so bad, it's so bad. So how it impact on the animal model? For that, we have used the hypoxic model as well as the rotinone model. So what happened, the hypoxia is also caused the protein aggregation, and PD-like model generated under the influence of rotinone. So what we have done, I can show you in the next slide. First, I just want to give you this hypoxia. This is a very common diagram that, okay, it's caused the misfolding of the protein, and this misfolding of the protein can be rescued by HSP 70 and 90 and CHIP and parking. So it can either protect the protein or degrade the protein under the influence of the ubiquitin E3 ligases. So here, what I wanted to tell you that in the left and right panel, in the left panel we have done the hypoxia experiment under the desiccator, and we have dropped down the oxygen partial pressure of oxygen from T1 to 8% through a sequential process. And this took us 30 days because, you know, I cannot drop down the 21% to 8% oxygen. It required some acclimatization. And for acclimatization, what we have done, we have just dropped the oxygen tension like 21% to 20% and so on and so forth, till 8%. So in this way, we have generated the acute hypoxic model. We have generated the chronic hypoxic model. In the left-hand side, in the right-hand side, we have intraperitoneally injected the rotenone to the rat in order to develop the PD-like symptoms. And here we have successfully generated these two models. And then we thought, now what would happen if we can use some biomolecules and check whether they are reversing the effect of protective proteins or reversing the effect of UPS machinery if it is in the degradation pathway. So these biomolecules we have taken in consideration: sesamol, naringenin, quercetin, and curcumin. These three are flavanones, and the curcumin is the phenolic compounds. So we have used these four biomolecules for our study. So I can show you here. You can see this is the H&E staining, and in the right-hand side is the silver staining. And you can see it here, normoxic, acute, and chronic condition. And after that, we have taken the acute and chronic in the presence of naringenin and curcumin. Quercetin, sorry. You can see it here in the acute condition. In the chronic condition, there's a lot of neuronal death in the cortical region as well as in the hippocampal region. And if you can correspond it with the silver staining, because the silver staining is a technique through which we can understand what is the level, what is the how many neurons are dead. So you can see it in the chronic condition that there is a cell death. Indeed, there is a cell death in the H&E staining, while in the silver staining, you can see the this spots which is depicting that, okay, there is indeed neuronal loss. But the moment we have added the naringenin and quercetin, these black dots are disappeared, which means that yes, these two drugs, these two biomolecules are able to reverse the hypoxic effect. Similarly, we have used the PD-like model using the rotenone, and you can see it here that there is a lot of neuronal death in the striatum and substantia nigra region. This is the corresponding blot of the silver staining. And again, the moment we have added the sesamol and naringenin, this process is, lethal process of this degradation process is reversed. Then we check the level of the thyroxine hydroxylase, and you can see it here. Under the influence of rotenone, we have successfully, we have shown that in condition of rotenone, there is a lot of loss of TH. TH is the precursor of dopamine synthesis. And the moment we have added this sesamol, we can rescue it. And here in the right-hand panel, you can see this beautiful immunofluorescence photograph, where what we have done, we have replicated the same experiment in the IMF, and you can see it that under the treatment of the sesamol and naringenin, we can find that neurons are going pretty well with control to the rotenone. Likewise, we have taken so many different protective proteins like HSP90, HSP70, PARC and CHIP, and the same thing we have replicated in the rotenone model. And everywhere we have found that yes, this quercetin and naringenin and sesamol and naringenin are rescued the protective protein levels. The same is done with this PARC and CHIP and DJ1, because these three proteins are very important, because these three proteins are working as a molecular chaperone as well as the ubiquitin E3 ligase. And so we have successfully shown that under the treatment of rotenone, these proteins are dropped down. But the moment we have added the sesamol and naringenin, this is again back to the control level. The same is done with this hypoxia, and we thought that, okay, if there is a decline in the protein production, it is going to the apoptotic pathway. And for that, what we have done this caspase-3 and caspase-9 expression. Caspase-9 is the initiator, whereas the caspase-3 is the executor. And we have seen that indeed, under the influence of different drugs in the hypoxic model as well as in the rotenone model, the cleaved caspase is completely gone. Likewise, here you can see there is no cleaved caspase in case of hypoxia in the red box, if you can concentrate on the cis-small naringenin in the both the condition caspase 3 and caspase 9, and likewise in the chronic condition and under the treatment of this drug. Then we thought if this is apoptotic pathway, whether it's going through the ubiquitination process or not. And for that, what we have done the ubiquitin experiment using the same cell lysate, and we have seen that under the treatment of drug, these two polyubiquitin chain, so please concentrate on the lane number 3 and 4 for the left-hand blot and the 3 and 4 in the right-hand blot, and you can see that indeed the 3 and 4 is drastically dropped in case of the drug treatment. Means if you can take home this, take home of this slide is under the influence of protective molecules, the polyubiquitin chain is gone. So what does this mean? Polyubiquitin is related to the destruction of protein, but the moment we have added the biomolecules, we have rescued the polyubiquitination chain. Then we have looked on the muscles, because PD-like model is a neuromuscular disorder, and that is why we check this under the with the you can see it here, this immunofluorescence staining of the muscles, and you can see it here how it is rescued with the help of cis-small and naringenin. And this is the Western blotting in the D panel. This also advocated the same thing, that the moment we have added the cis-small and naringenin, the protective protein levels in the muscle is upregulated in control with the rotenone. Then we have thought that, okay, if this is happening, whether the Parkin and CHIP are means like a staining is interacting. So first we have checked the individual staining of the Parkin and CHIP staining. And you can please concentrate on the left-hand panel C and D and right-hand panel C and D. This is only the rotenone. So you can see that rotenone, the cells are dying like anything, but the moment we have added the cis-small, the Parkin level as well as the CHIP level is upregulated. This the same thing we have shown you in the previous slide in through the Western blotting. Now come to the TH staining and the ubiquitin staining. Again, the same thing, that in under the influence of cis-small, it is upregulated, the TH production, as well as the ubiquitin staining is dropped down, because we don't want ubiquitin. If there will be more ubiquitin, there will be more destruction of protein. So here we have saved the neurons. That's what we have saying. This is the complete reversal picture, like in rotenone and cis-small in the left-hand panel, the neurons are growing well, whereas the moment we have checked through the ubiquitin antibody, we have shown that, okay, cis-small is dropping down the ubiquitin staining, because you can see it in rotenone, there's a lot of ubiquitination. There is a lot of green fluorescence, which depicted that neurons are dying. But the moment we have added the cis-small, this ubiquitin staining is almost gone. Then we asked the question, if ubiquitination is so important, whether it's following the apoptotic pathway, and for that we have done the double staining, and we have confirmed, yes, ubiquitin and caspase 3 are interacting, and thus it is following the apoptotic pathway. The same caspase 9, caspase 3 staining we have done, and we have found the same thing under the cis-small. The cleaved caspase level go in the down panel, but in the immunofluorescence, we have shown, yes, there is a no means like a sign of apoptosis. So these papers we have published in this brain research as well as the back to the neuroscience paper, and it get a lot of attention in the scientific fraternity. Then we thought that, okay, if there is any crosstalk between the AD and PD, so what we have done, we have I can show you here that we have done the pulse stage experiment. The beauty of pulse stage experiment is it can say you what is the rate of biosynthesis of a protein as well as the degradation. So here in the left-hand panel, in the right-hand panel, we have used the CHIP as well as Parkin. And again, we have shown through the pulse stage experiment that indeed under the influence of CHIP and Parkin, the A beta level is dropped down during the production of protein, during the production of A beta. So exogenous CHIP and Parkin enhances the degradation rate of the A beta 1 to 42. Then we thought what would happen if we combined with, when we combined, we have also shown the same effect, that it is accelerating the the A beta, sorry, it is degrading the A beta level drastically when the both cognates, CHIP and Parkins are present. So to conclude my work, here you can see that hypoxia is causing the mitochondrial dysfunction and also this misfolded protein, but it can be rescued using the flavonoids, as well as it can also help in the neuroprotection. Again, the same thing, cismol and naringenin can cause the cell survival and cause the neuroprotection, whereas these PD-like model can having very devastating effect like the Ross generation, as well as the accumulation of different proteins, UPS dysfunction, cytosolic accumulation of the proteins, loss of dopaminergic neurons, but that which cause the Parkinson disease or neurodegeneration. But the moment we can add the cismol, we can rescue this. In the third conclusion, that yes, ubiquitin is very important, but at the same time, we have to think how to utilize this ubiquitin pathway for the therapeutic steps. So these are some of my papers, which is published. We have published in the 2021. So I'm really thankful to all my wonderful PhD students who has done a splendid job in my lab. And these are the different ongoing projects and the fundings. So I would like to thank all my former PhD students and current PhD students who is doing a exceedingly nice job. Thanks for your attention, and I'm really sorry because of me, it's a lot of inconvenience occurred. So once again, I apologize for my this thing. Thank you. If you have any question, please ask.
Thank you very much, Dr. Kumar, for such an informative and exciting lecture. There are a few questions in the chat box. Can I ask?
Yes, madam, please.
Dr. Bhupendra Kumar wants to ask why these 4 molecules you have taken, and how did you ended up in selection of these 4 molecules? See, there are many other bioactive molecules which can be used in different conditions, including cancer. Is there any particular reason that you have chosen these 4 biomolecules?
Madam, first and foremost, we wanted to check the action of flavonoids. So these flavonoids are completely in my system is not very known, especially if I can talk about the cismol. In 1978, there are some studies showing that cismol is important for the neuroprotection. But since then, nobody has worked extensively on this molecule. So this was one reason why I have taken the cismol. Regarding the quercetin and this naringenin, quercetin is HSP72 specific blocker. So we wanted to check whether this protein molecule, if it's a protective protein HSP17, if it's a block, how this molecule would behave. Curcumin, you can say, ma'am, because curcumin is very common and it's not studied. So I thought that why not we can also take the curcumin for our study in order to understand what is the impact of this phenolic compounds on our neuronal system.
Thank you. So what is the mechanism by which these molecules are acting?
Madam, the mechanism is right now, we have shown you all these molecular pathways. We have taken all the signaling pathway because, our target was how the upstream targets of a protein and downstream target of the proteins can functional. And that is why we have taken so many different signaling pathways.
Sir, is ROS the main culprit?
Ma'am, ROS is the main culprit for everything. But here we are more concentrating about the UPR, unfolded protein response. We are working on the UPS, ubiquitin proteasome system machinery, how it is recruiting the lysine residue or so that it can be attracted by the ubiquitin and it can make a polyubiquitin chain and it can degrade the protein. So here our main focus was to understand first chaperonic pathway, UPS pathway, apoptotic pathway, and then other signaling pathways.
Thank you so much, sir.
Thanks a lot, ma'am. Is there any question,
ma'am? I think no more questions. Thank you.
Okay. And I'm really sorry, Manjula madam, please excuse me. So now I can leave.
Yeah, sure. Thank you, sir. Thanks a lot.
Thank you.
It is with the
Thank you so much. So the next speaker is Professor Ravi Shankar Akundi. Professor Akundi is an assistant professor at the Neuroinflammation Research Lab, Faculty of Life Sciences and Biotechnology, South Asian University in New Delhi. He is the winner of the Barnstable-Brown Obesity and Diabetes Research Day Award and recipient of the American Heart Association Heritage Fellowship at Yale University, United States of America. Brain inflammation is a common occurrence in neurodegenerative disorders, and therefore, Professor Akundi's presentation titled Extracellular ATP-mediated cyclooxygenase 2 modulates inflammation in the systemic and neuronal disorders will elaborate on the manner in which exogenous ATP binds and modulates inflammatory reactions during neurodegeneration. Professor Akundi, a very warm welcome to you, sir. The floor is all yours.
Professor Akundi, you are co-host now. You can share your screen.
Hello, good afternoon, everyone.
Good afternoon, sir.
Good
Welcome.
afternoon, sir. Thank you so much for adjusting the time, sir.
Are my slides visible?
Yes, yes, they are, sir.
And are they moving? Is my slides moving?
No.
Your slides are visible to me, yeah?
But not moving. Yeah, moving now. Let's do it again.
I'm doing full screen. Is it moving in the full screen?
No, no, no. Full screen is not moving. This way it's moving.
Oh, okay.
Could you please move one slide?
In a non-full screen, I think it is moving.
Yeah, yeah, it's moving. This way it's moving. Yeah, yeah.
Now I have made full screen. Can you check?
Yes, yes, it's working.
Is it moving? It's working? Okay. Then I'll stick to full screen. Right. Good afternoon, everyone. Thanks a lot, Dr. Imran and Dr. Manjula Suri, Professor Suman Jain, and everyone for making me a part of this symposium. I'm really honored to present my work here. This is my work from South Asian University, which is located currently in Chanakyapuri. We'll soon be moving to a permanent campus in Maidangari in the later half of this year. So the talk for my talk is extracellular ATP-mediated FOX2 modulates inflammation in the systemic and neuronal disorders. Now, the keyword here I have is inflammation. So what is inflammation and where does it occur? If we look at inflammation, it is predominantly present in most of the disorders that we know, starting from acute infections like the bacterial infections, all the way to metabolic disorders, cancer, and neurodegenerative disorders. If we just look at the acute infections like the bacterial infections, and we just look at the neonatal infections, in the case of South Asia, there is almost a neonatal mortality of 48 per every 1,000 live births. And among those who survive, for example, Japanese encephalitis virus or other kinds of bacterial meningitis, many of them suffer from long-term cognitive impairment. South Asia and especially India is also one of the countries where the highest amount of incidence.
Apologies, Sorry, sir. Are you moving your slides?
Okay, so it's not moving. Then I'll keep it in
Yeah, it just
non-screen.
moved. It just moved. We're seeing a diagram right now.
Yeah, now I'm not doing
Inflammation.
in full screen mode. Yeah, I won't do full screen mode. I'll
Thank
just
you. Thank you.
leave it like this. Yeah. South Asia is also where you have the highest incidence of traumatic brain injury and also cerebrovascular diseases like stroke. Again, in these disorders, in these diseases where you have injury to the brain, those who survive, they suffer from a long-term morbidities. Inflammation is also seen in neurological disorders, neurodegeneration diseases like Parkinson's disease. We just heard about it in Alzheimer's disease, and also in other disorders like depression, where there is a component of inflammation or a chronic inflammation. This chronic inflammation is also a component in metabolic disorders like diabetes. And in 2011, Weinberg and Hanahan have included inflammation as one of the hallmarks of cancer. It's as an enabling characteristic. So these things shows that inflammation is at the core of many of these disorders, both in the acute as well as in the chronic conditions. And therefore, targeting inflammation helps us to find a therapy for many of these disorders. As such, inflammation has a lot of role in normal physiology. It helps in the elimination of the injury, wherever the site of injury and what is the cause of the injury. And at the same time, it will help in the removal of all the dead cells and necrotic cells through a process called phagocytosis. And this is predominantly mediated by the innate immune cells like macrophages in the systemic system and microglia in the central nervous system. A major component of inflammation is there has to be a resolution towards the end. That means where the tissues can be repaired. And failure of this resolution is where inflammation becomes from acute to a chronic or becomes persistent. While these are the cardinal signs of inflammation, these are all mediated by various chemical mediators which come from either the cells or in the vascular system. And among these mediators, what I'm going to predominantly talk today is about the eicosanoids, in particular prostaglandins. Now, if you look at these eicosanoids, they are derived from the 20-carbon arachidonic acid, which is present within the phospholipid membrane. And whatever is a stimulus, it leads to the activation of phospholipase A2, which eventually causes the release of the arachidonic acid from the plasma membrane. Now, this arachidonic acid undergoes a cyclooxygenation and a peroxidation reaction, which is mediated by the enzymes known as cyclooxygenases. These cyclooxygenases add a cyclic oxygen molecule at the C9, C11 carbon of the eicosanoids and eventually cause a cyclization reaction. It is also a source of free radical because there is a presence of a free electron which will now remain in the system. So it also is a source of non-mitochondrial free radicals within the cell. The end product of this cyclooxygenation reaction is the PGH2 or prostaglandin H2, which serves as a precursor molecule for a variety of prostaglandins, including prostaglandin E2. So these precursor molecules are synthesized by independent prostaglandin synthases. There are various forms of these syntheses. There are cytosolic ones and there are microsomal ones. This pathway that I told you is just one of the pathway which is shown in these rectangular brown squares here towards the synthesis of prostaglandin E2, from where you can see arachidonic acid is converted to PGH2, and from there the PGE2 synthase leads to the synthesis of prostaglandin E2. This prostaglandin E2 has a variety of role in vasodilation, hyperalgesia, fever, diuresis, and immunomodulation, among many other roles that it has. This PGH2 also serves as a starting molecule for the synthesis of prostaglandin D2 through a PGD2 synthase, prostaglandin I2 or prostacyclins, thromboxanes, and PGF2. So all of these have different roles. Some allow vasodilation, some cause vasoconstriction, some allow platelet aggregation, some inhibit platelet aggregation. So it is a combination of all of these molecules which help in the early phases of inflammation, and some play a role in the later phases of inflammation or during the resolution steps. In addition to cyclooxygenases, there are other group of enzymes known as lipoxygenases, which help in the synthesis of leukotrienes. And this is a separate pathway, which we are not, I'm not going to concentrate in this current talk here. So here in my talk, we are mostly interested in the role of the cyclooxygenases, which eventually lead to the synthesis of this prostaglandin E2. Now, so since this PGE2 is involved in multiple of these acute and chronic inflammatory conditions, it becomes very important to regulate the synthesis of or the activity of cyclooxygenases. All cells have two different types of cyclooxygenase isoforms. There is COX1 and COX2, which are around 70 to 72 kilodalton in molecular weight, which are responsible eventually for PGH2 synthesis and eventually to PGE2. When they were discovered, it was found that COX1 generally does not change with response to stimulus, so it was considered to be as a housekeeping enzyme, which remains more or less constant in different cell types under varied stimulatory conditions. Whereas COX2 was found to be activable by various stimuli, particularly by cytokines and other mediators. So this became an inducible form. So we have COX1 as a housekeeping, was considered as a housekeeping protein, whereas COX2 as an inducible protein. And this COX2 as an inducible gene, which leads to the synthesis of high levels of COX2 whenever there is inflammation. And this is particularly true for microglial cells and macrophages. Before the actual role in inflammation, there have people from antiquity, people have been using various types of anti-inflammatory drugs. And one such anti-inflammatory drug traditionally used comes from the willow plant, which had been used by ancient Mesopotamians and Egyptians for treatment of pain and inflammation. In fact, there are also uncited reports that Hippocrates had advocated willow leaf tea to women to ease the pain of childbirth. Documented reports of compounds isolated from the willow bark comes from 1828 when Joseph Buchner extracted the active component as salicylin, which was chemically synthesized by the French scientist Charles Gerard in 1853. And one of the earliest clinical trials of salicylin was done as long back as in 1876, which was also published in Lancet at the time, as being capable of relieving fever and joint inflammation in patients with rheumatism. In 1897, Felix Hoffmann added an acyl group to this salicylin or salicylic acid, which reduced the irritant property of salicylin. Thereby, it became a more attractive as a drug, which Bayer eventually patented and marketed in 1899 as aspirin. Aspirin, within half a century, entered the Guinness World Records for being the most frequently sold painkiller, even before its mode of action was discovered by John Wayne in 1971, for which he also received the Nobel Prize. In addition to aspirin, once its commercial value has been discovered, many other anti-inflammatory drugs have come into the market, such as paracetamol, ibuprofen, indomethacin, naproxen, and many more are there, which I have not listed here. All of these are generally grouped under non-steroidal anti-inflammatory drugs. As of 2010, I don't have the latest data. The turnover for aspirin itself was 766 million euros. Now, while these are good for fever, inflammation, and many acute conditions that we had discussed in the previous slides, are these any of these non-steroidal anti-inflammatory drugs useful in chronic inflammatory conditions where there is persistent levels of prostaglandin E2 present within the system? Since COX2 plays a major role in neurodegenerative disorders, aspirin had also been tested in neurodegenerative diseases like Alzheimer's disease. And there are many clinical trials that one can see, which have, most of these have been completed in the 1990s to 2000 or 2005. Many have been completed or they have been stopped in between the studies for lack of various data points. The end result of all of these studies showed that they may be effective, but there is no change as far as cognitive decline is concerned. So NSAIDs do not help in ameliorating reducing the cognitive decline that people from Alzheimer's disease suffer from, nor do they change the progression of the disease. So as seen in one of these studies, long-term NSAIDs can reduce the risk of AD, but then they have to be started well before the onset of dementia. Other studies showed that there is no NSAIDs have not been useful in the treatment of AD. So there have been different types of NSAIDs like ibuprofen, naproxen, salicylate, and other things. Even drugs like curcumin have also been tested. A review of all of these clinical trials shows that while there may be some uses of NSAIDs in preclinical studies as being associated with lowering the risk of Alzheimer's disease, as far as randomized trials are concerned, they have been not effective. And one of the major reasons for these things, also a major component, is also a large number of patient dropout happened during these clinical trials, especially among the healthy volunteers who were taking NSAIDs. And that is because of the frequent gastrointestinal complications on daily usage of this aspirin. This linkage between daily use of aspirin and gastrointestinal complication kind of reduced in statistical numbers for many of the clinical trials that were conducted. Another chronic inflammation that has been widely studied in terms of clinical trials is the association of whether aspirin can have an effect on the progression of cancer or the risk of cancer. Again, this is just for aspirin and not other NSAIDs, and these have been done at many places. But as you can see, many have withdrawn or they are no longer recruiting. Again, the major reason that comes across is the lack of numbers. Although for some studies, it has been shown that if aspirin has been taken before the age of 70, then there is a reduced risk of colorectal cancer, for example, or the urine PGEM, which is the marker for prostaglandins. It has been reduced in at least 10% of the individuals suffering from colorectal cancer. But many studies were terminated or were not recruiting, again, because, as we can see here, because of the severity of gastrointestinal disorders. Some have 7% had severe GI disorders, but almost nearly everyone, 26 out of 27 patients, they had not serious but adverse events associated with gastrointestinal disturbances. Now, why such a huge number of GI disturbances when NSAIDs were used? This comes from the downstream product prostaglandin and its activity. This prostaglandin E2 acts through 4 different types of, can act on through one of the 4 receptors, which are termed as prostanoid receptors or EP receptors, EP1 to 4. These are associated with different G proteins, and their role, and they are present throughout the systemic body, including and in the brain too. And these are associated with various gastrointestinal processes like the contraction of the smooth muscles in the gut, the release of the gastric juices. They are involved in the renal system, in the renal filtration system. They are involved in the contraction of the bronchial muscles, and relaxation of the various muscles in the reproductive system. And they are involved in nearly entire system, in almost all physiological functions that we know of. Therefore, inhibiting this PGE2 or COX2 led to many of these side effects that are associated with functions of PGE2. As I told you, there are 2 isoforms of this enzyme. And since COX1 was considered to be a housekeeping enzyme, and NSAIDs can act on either of these enzymes, what was thought was since inflammation is because of a predominant increase in expression of COX2, whether selective COX2 inhibitors would be helpful. Thereby, one would just stop the activity of the COX2 enzyme, the inducible form, whereas the housekeeping roles of the prostaglandin E2 and the basal levels of prostaglandin E2 within the body would be maintained by the COX1. And therefore, a series of inhibitors specific only to COX2 have been made, which are known as the COXIBs. However, usage of COXIBs were also associated with severe risk factors. And in fact, one of these drugs, known as Vioxx, was marketed by Merck, whose sales were increased because they had shown very good effect in rheumatoid arthritis patients. But then many of these patients suffered from severe cerebrovascular and cardiovascular disorders like stroke and heart attacks, including many people who also died because of usage of Vioxx, because of either heart attack or stroke. And as this report says, rofecoxib should have been withdrawn many years ago, since these long-term studies had not been conducted on it. So this tells us that even though COX-2 is an inducible form, and there is a COX-1 which is not inhibited by these specific COX-2 inhibitors, nevertheless, inhibition of either of these enzymes had led to severe side effects, including mortality and morbidity. So we need to have alternatives to reduce the levels of PGE2, which is the inflammatory mediator, but it should not be associated with the direct inhibition of the cyclooxygenase enzymes. And in this search, we came across ATP as one of the molecules. What ATP? Well, just going to describe. This is what we know as the inflammatory condition. So you have a resting cell, resting innate immune cell with a macrophage or a microglia, which normally expresses COX-1 under basal conditions. And whenever there is an injury, whether it is a bacterial infection or a traumatic brain injury or ischemia or neurodegeneration, acute or chronic, any kind of insult that is given to these cells, then cells get activated, which basically is the induction of the COX-2 gene and increased expression of COX-2 protein, thereby increased levels of prostaglandin E2. And this is what was being considered as the inflammation in the system. Now, tissue injury is also associated with the cells which are damaged. And once the cells are damaged, which means once the cell membranes are broken up, then these cells with their whose membranes are broken, their cellular contents or the cytoplasmic contents are now out in the extracellular milieu. So their cells are rich in a lot of things. The cytosol is rich in a lot of ions and molecules. Now, all of these things are within the extracellular milieu. What effect do these have on this in this process of inflammation? And one of the major molecule that is present within the cytosol is the ATP itself. Cells usually have in high millimolar 5 to 10 or even higher millimolar concentrations of ATP depending on the cell type. And now this ATP is now within the extracellular milieu. And we propose that this cell death, the release of these components, especially ATP, this serves as actually the second hit in the inflammation. So while the infection and ischemia, whatever is the direct, the cause is the first hit, which is causing the activation of the cells. This in fact is only the initial stage or the first stage of inflammation, but the actual inflammation comes from the increased release of ATP by the dying cells, wherein this ATP acts on the P2 receptors of these innate immune cells, and through the PT receptors leads to increased expression of COX-2, thereby increasing the levels of prostaglandin within the system. As a proof of concept, we then tested it. And as you can see here in primary human monocytes, LPS, for example, leads to an increase in the level of prostaglandin to around 15,000 picogram per ml, as you can see. But if you add ATP together with it, we are adding it exogenously in the culture. These are in cell culture model. So when we add ATP into the culture media, then you can see from 15,000, it has gone up to 30,000, twice the amount of prostaglandin E2 has been synthesized or released into the media. The same is true for macrophage cells, ROD264.7 cell lines. You can see there is up to 10,000 with 10 nanogram per ml LPS. And when we add ATP up to 1 millimolar, then it is almost fourfold increase in the amount of PGE2 that is released in the extracellular milieu. So this, same with the 100 nanogram LPS, you have, and with ATP, there is a 4 to 5-fold increase in the amount of PGE2 that is actually synthesized by these cells. And this comes from the increased expression of COX-2, so which we can see in the Westerns here. There is not much change as far as the levels of COX-1 protein is concerned. But when we look at the levels of COX-2 protein, from a minimal expression in the control cells to there is an increase with LPS, and with LPS and ATP, there is nearly twice the expression of the protein. So this tells us, this shows a proof of concept that when you have ATP in the extracellular milieu, then this causes the innate immune cells to increase the amount of prostaglandins that they synthesize by 2-fold or 4-fold or depending upon the cell type. And This is also true in the case of central nervous system. These are primary rat microglial cultures. And as you can see with the LPS alone, 100 nanogram per ml LPS, that is around up to 1000 picogram per ml PG2 that is released in the media, which increases with increasing concentrations of ATP. We can see with 1 millimolar ATP, there is nearly 8-fold increase in the amount of PG2 that is synthesized, which is also seen here in the amount of COX-2 that is expressed. Not only just COX-2, but including the prostaglandin E synthase is also increased. Further, to look at the proof of concept in a different way, we treated cell line. We took HeLa cells and we treated them with doxorubicin, which at its LD50, so thereby ensuring that these cells die, and we can see that there is an increased amount of cell death. And then we measured the amount of ATP that is released in the media in response to the cell death, and there is an increase in the amount of ATP corresponding to the increase in the amount of cell death that we see in response to doxorubicin. We use this media essentially as conditioned media, which is basically the media of HeLa cells which have been treated with doxorubicin. We use this media on our macrophage cells, and you can see that if we use regular media from HeLa cells which are under normal conditions, so there is not much change in the COX-2. But when we use the media that is obtained from HeLa cells which have been treated with doxorubicin, then you can see there is which is considered as a conditioned media, there is an increase in the levels of COX-2. And further, if you add other insults like LPS or other things, so there is further increase in the amount of COX-2 expression. So this essentially, this conditioned media essentially is the ATP is a contributory factor here, and this is the LPS plus ATP. So it demonstrates the same effect as we see when we exogenously add ATP into the culture, or we use the ATP that had been released ATP along with other components that has been released by the dying cells. Not only the cells show increased expression of COX-2, they also show increased activation, which we can measure in terms of migration. Since these macrophage cells are migratory in nature, we used a transfer assay system where you plate the cells on the top of a transfer filter, and then you measure the amount of cells that migrate from the top of the chamber to the bottom chamber. And as you can see, LPS causes activation, and with ATP, exogenously added ATP, there is a significant increase in the number of migrating cells. The same is seen in the case of conditioned media, where the cells are treated, if they are treated with just media from normal HeLa cells, there is not much migration. But once you add the conditioned media, that is the media that comes from HeLa cells which have been treated with doxorubicin, the dying cells, when you add them, there is an increase in the amount of migratory capacity by these cells. So this shows that not only the COX-2 levels, but also their activation, including their migration, is enhanced when ATP is present within the in the media, either exogenously or coming from dying cells. So this shows our proof of concept that exogenously present ATP acts as a major inflammatory modulator. That ATP is a universal stress signal. This has been well demonstrated. In fact, it is considered as a damp molecule. It is associated as a danger-associated molecular pattern molecule, and not just in animal systems. In fact, even in a plant system, it has been considered especially when herbivores and pathogens, they eat the leaves or create wounds in the plants. Then there is a release of ATP from intracellular in millimolar concentrations where the cell's ATP is present. It is now released out into the extracellular milieu. And this ATP, it can act on these purinergic receptors, which have also been demonstrated to be present in the plants, which are known as DON receptors, or does not respond to nucleotides. So these receptors bind to ATP and thereby increase this inflammatory signal. Generally, the ATP that is present in the extracellular milieu, it is usually under normal healthy conditions, is present in very low concentrations because of the cell surfaces are rich in what are known as ectonucleotidases. These ectonucleotidases are responsible for hydrolyzing the ATP. Since ATP is a danger-associated molecule, so that means if there is increased concentration of exogenously present ATP, then that serves as a signal that some kind of injury has happened in that vicinity, which will attract the macrophages and create the inflammatory situation. So therefore, in general, cells express, many cells express a large amount of ectonucleotidases on their cell surfaces. And the role of these ectonucleotidases is to basically hydrolyze these nucleotides. We have CD28 and CD39, which hydrolyze ATP down to AMP, and AMP is further hydrolyzed down to adenosine by CD73. This adenosine can enter back into the cells through nucleotide transporters, and thereby the cycle completes. The cell, again, if the cell is not injured and is perfectly fine, then it will take back the adenosine and the pathway is completed. Alternately, all of these nucleosides and nucleotides, both adenosine, ADP, ATP, all of them also have receptors on the cells to which they can bind and activate downstream signaling pathway. Conventionally, ATP and ADP act on purinergic receptors, which are called as P2 receptors, and they have been shown to be more as pro-inflammatory, including in our studies. And the adenosine binds to a different kind of G protein coupled receptors called as adenosine receptors. And these are generally considered to be anti-inflammatory, but these are only, there's a lot of debate among all these receptors and variation in data also. And this we can also see here. If we have hydrolyzed components of ATP, then the amount of COX-2, NICA is essentially a stable form of adenosine, which does not induce any COX-2 within the primary rat microglia. Again, because cells can, when cells die, they release all the components, whether the effect that we are observing is specifically coming from ATP or not, or whether some other component within the cells, where if they are responsible for increased inflammation. To test that, we also stimulated cells with what is called, an enzyme called APRC, which is basically hydrolyzes ATP down to adenosine. And as you can see, with LPS and exogenously added ATP, there is a strong expression of COX-2 in the microglia. And when we treat the cells with APRC, which basically hydrolyzes the ATP down to adenosine, the amount of COX-2 expression is reduced back to control levels. So this tells us that among all the cellular components that have been released when they are broken, ATP is probably the one that is playing a major role as far as COX-2 synthesis is concerned. And so we can reduce this effect when we use APRC. We also tested this in an animal model. We took mice which have with these conditions treated just with saline, or mice which have been treated with LPS at 8 microgram per gram body weight for 3 hours, and APRC was also injected in some of them. These are intraperitoneal injections. And after 3 hours, then we collected the peritoneal fluid. And as you can see in mice, in control mice, there is a very little amount of peritoneal ATP in general, but this increases almost up to 7 to 8 fold in mice which have been injected intraperitoneally with LPS. In contrast, when the mice are also injected with APRC together with LPS, then the amount of ATP that is present is very low. And this you can also see in the COX-2 in mice which have been treated with LPS. There is a strong increase in the expression of COX-2 within the peritoneal macrophage cells which have been isolated from these rats. And when they have been treated together with APRC, then the amount of COX-2 expression is reduced. Also, there is no change as far as COX-1 expression in these macrophages are concerned. So this tells us that the COX-2 is an inducible form, and APRC can actually reduce the levels of inflammatory COX-2. We also cultured these cells, and this is, for example, to look for the markers. CD11b is one of the markers for these macrophages. So with LPS, there is a strong increase in cells expressing CD11b and COX-2. And this is reduced in cells which have been treated with suramin. Suramin is a broad spectrum purinergic receptor antagonist. It binds to several different types of P2 receptors, and thereby one can see that the expression of COX-2 is reduced, as well as the amount of CD11 positive cells are also reduced in the peritoneum. We look at the amount of ATP that is released. It's strongly released in response to LPS. There's not much change when the rats have also been injected with suramin. We injected with 5 microgram per gram body weight intraperitoneally. But we can see that there's a significant reduction in the amount of PG2 within the peritoneum comparable to control levels. And the amount and the number of F480 positive macrophages are significantly reduced within the peritoneum of animals which have been co-injected with suramin along with LPS. So this tells us the inhibition of P2 receptors abrogates LPS-induced inflammation in the peritoneum. These extracellular nucleotides, they act on purinergic receptors, and there are around 15 known purinergic receptors so far. Among these, 7 are ion channel based receptors, P2, which are known as P2X1 to X7. And there are 8 of these which are G protein coupled, which are known as P2Y receptors and named P2Y1, Y2, Y4, Y6, Y11, 12, 13, and 14. We selected a few of these purinergic receptors, those which have whose at that time whichever were available as far as their complete sequences are concerned, and for which antibodies and specific agonists and antagonists were available, and which have been previously reported in literature in one or the other studies. So we use these purinergic receptors in our studies, and we saw that these macrophages, for example, they predominantly express the X4, X7, and Y6. Same in the case of microglia also, they express X4, X7, Y6, and among the ones that we have tested. We also use specific agonists and antagonists and to further identify which of these purinergic receptors plays a role in exogenous ATP-mediated COX-2 induction. This is again a proof of concept in the case of primary rat microglia. The treatment with suramin, if you culture the primary microglial cells and LPS and ATP together, shows a strong expression of COX-2, which is seen in this green color, which is now reduced when cells are also co-treated with suramin. So among the various agonists and antagonists among that which we have screened, in the case of primary microglia, we can see UTP, which is for P2Y4, and UDP, which is for P2Y6. They both have shown strong increase in the release of PGE2 and BZ-ATP, which is a stimulator for P2X1 and X7. It shows a strong increase in the levels of COX-2 also. So this shows that X7 and Y6, among others, are predominantly expressed in primary microglial cells and also probably involved in ATP-mediated COX-2 expression. So we further came down to these by using other known agonists and antagonists. And of course, these are chemical screenings, and there is always a chance that some of these inhibitors may bind to other receptors also, to which they are not, the specificity may not be very strong. In the case of raw cells, macrophage cells, again, we further confirmed that the P2X7 receptors are involved and P2Y6 receptors are involved by using the same strategy, by stimulating the cells with the specific agonists and antagonists in various combinations, and we came down to the same conclusion. We also tested that, for example, this Y6 antagonist also reduces the COX-2 level when cells are also treated with the conditioned medium obtained from the cells which have been treated with doxorubicin. So our studies therefore shows that what we know as inflammation which we conventionally thought is inflammation is just the first hit, but what we actually observe within the system is a second hit, which is basically coming from the ATP that has been released from the dying cells. So the actual inflammation that is observed is much higher, and one can reduce the intensity of this inflammation. One can bring it down to clinically manageable levels, perhaps by using these P2 receptor antagonists, because these P2 receptors antagonists would basically block the P2 receptors, and therefore it will reduce the excessive synthesis of COX-2, which is coming from the P2 receptor downstream of the P2 receptors. So what the direct insult itself causes the activation of TLR receptors, for example, and downstream activation of COX-2. So that is what would remain as the first hit, and that is what one can bring down the level of inflammation. So although these P2 receptor-based anti-inflammatory drugs will not completely bring it down to the resting phase, but they would significantly reduce the effect of the second that is coming from the second hit. However, there is a lot of roadblocks as far as designing a good P2 receptor anti-inflammatory drugs is concerned, because as I told you, there are 15 different types of receptors. So there is inherent diversity within these receptors. And therefore, also there is a lot of receptor compensation that can happen, or there can be differential expression on different cell types and different effects. In one cell type, it can be actually causing an increased inflammatory response, whereas in another cell type, it can be actually reduced response. For example, P2X4 is essentially serves as a marker on the neurons for them to be phagocytosed, whereas on the microglia, the P2X4 essentially serve as a pro-inflammatory mediator. So this one has to consider the various differences between these receptors and how to actually come across the to choose a perfect antagonist to address a particular disease. In addition, one also has to see that injury will release all kinds of nucleotides, and then there are a lot of ectonucleotidases. So we have all kinds of things in the extracellular milieu, from ATP all the way to adenosine, and each of these can have different effects on the inflammation, downstream inflammation. One example I'm showing here. This is where we had read the pups in hypoxic condition. So these are from P3, they were kept in a hypoxic chamber until P12, together with the dam, of course. And one can see that this hypoxia causes a reduced myelination within the brain. As we can see, compared to normoxic animals, there is a reduced amount of myelination that one can see. Here, the anterior commissure is here, and the corpus callosum. There is a reduced amount of myelination. But when these animals have been treated with caffeine, and caffeine is one of the adenosine receptor antagonists, so which blocks the adenosine receptors. When caffeine is given, it is administered in water, and it is given to the dams. Although the dam takes it through the milk, it goes to the through the lactation process. It goes to the pups also, and there is a significant amount of caffeine that can be seen in the pups also. In pups which have received this caffeine, this effect is almost gone. So if actually we see a large increased ventricle size in hypoxic animals, so which is called as a ventriculomegaly, and which is essentially reduced in the case of pups which also received caffeine or the adenosine antagonist. Same is the case of myelination. In normoxic conditions, you have a good myelination. Hypoxia, there is a reduced myelination. And in animals which received caffeine, again, the myelination is restored, which we can also measure in terms of looking at the levels of myelin basic protein. So this shows that if we block the adenosine receptor, that is actually helpful in these animals which were in hypoxia. Whereas in other studies, we see that if we reduce the levels of ATP and bring it down to adenosine, then this adenosine is actually acting as through adenosine receptors, is acting as an anti-inflammatory molecule. So it is all context dependent, whether we are looking at inflammation, adenosine is perhaps good, but in the case of adenosine signaling is good, whereas in the case of hypoxia, inhibition of this adenosine signaling is probably better. Now, this model where we have seen that this ATP is increasing the amount of inflammation in the cells, is this model applicable to any other conditions where cell death is also prominent? Because cell death is very common, not just for bacterial infections and traumatic injuries. It's common in other conditions also. And so in those conditions also ATP release occurs, and whether this model can be applied there. So one such model we had thought about is in the case of tumor cells. Now, when tumors cells are treated with either chemotherapeutically or radiotherapeutically, there is a large amount of cell death that happens. So this is coming from cells that are sensitive to the drug or the radiation. And so when these cells die, there is a large increase in the amount of ATP within the extracellular, or with what we call it as a tumor microenvironment. Although there is a large number of cells die, we also know that some of these cells do not die either to the radiation or to the chemotherapy. And these are what we call it as a chemoresistant cells. And the question is whether this ATP that has been released, whether this has any role in the tumor microenvironment. And what we had hypothesized is whether this ATP can bind to these P2 receptors, which might be expressed on these chemoresistant cells. And in case they bind to that, and if in case the chemoresistant cells express P2 receptors, then the ATP binds to them, and that will cause to increased induction of COX2 in these cells. COX2 has been shown as a mediator for metastasis in the case of tumors. So, and as I told you, in 2011, inflammation has been included as one of the hallmarks of cancer. And the way it mediates cancer is essentially by having cancer resurgence or recurrence, where the presence of cyclooxygenase affords the cells with migration and invasion properties. So we propose that the release of this extracellular ATP might be involved in increased migration or affording the metastatic properties to these chemoresistant cells. Again, as a proof of concept, we took 3 different cells. We chose a cervical cell line, HeLa, we chose a neuroblastoma cell line, IMR32, and we chose a breast cancer cell line, MCF7. In all the 3 models, cervical, neuronal, and breast cancer, we see that when you treat with doxorubicin, there is a significant amount of cells that die, which when you measure the amount of ATP that is released, which is also significant. We call it as a residual ATP because we are measuring the amount of ATP within the media 24 hours after the cells have been killed. I mean, after doxorubicin has been given. So within this 24 hours, the ATP can actually be hydrolyzed by ectonucleotidases, which are expressed by the cells. It can be hydrolyzed by various components within the serum, which since the cells are growing in serum-rich media, or it can be taken up by the cells through different pathways. So whatever is remaining is what we call it as a residual ATP. And therefore, the amount of residual ATP may be in smaller amounts. But then when we had used these as a conditioned media in our studies also. Here, which I'm showing in the next slide, here we are adding directly exogenously the ATP, and we can see if we have ATP within the media, it causes increased migration, here seen in the case of wound healing. So the wound is essentially healed when cells are treated with ATP. And again, we add a P2 receptor antagonist, this process is reduced. In all the 3 cell lines, nearly the process is inhibited. So this tells us having ATP in the tumor microenvironment helps in the cancer cell migration. Again, this effect is at different concentrations for each of the cell lines. So we picked up 1 concentration each for the different cell lines that we were studying. And again, migration was also studied because wound healing is, again, depends on the user and the way the wound is created and other things. So we also used a transfer acid as a confirmation, and we can see that when ATP is present, the black bars show that it increases the amount of migration or migratory cells. And when we treat the cells with suramin, the P2 receptor antagonist, or with APRC, which hydrolyzes ATP, both of these reduce the migratory capacity of these cells. So we see very few number of cells which are present. As I told you, we also use the conditioned media from the dying cells 24 hours after. So whatever is the residual ATP, although it is in lower concentrations, we use these as the conditioned media to check. And this conditioned media from the doxorubicin treated cells, for example, shows increased migration, and which we can block by using suramin. All the 3 cell lines we can see there is increased migration as well as invasion. Invasion is essentially the same assay we are using, but wherein in the transfer, in the migration, we are directly plating the cells or seeding the cells on top of the transfer filter. In the case of invasion, we add a layer of matrigel on top of the transfer filter. And on top of this matrigel, the cells are seeded. So the cells basically have to digest the matrigel and invade basically in order to come to the bottom surface or to the where the conditioned media is present. So both migration and invasion is seen by this conditioned media, and which we can inhibit by using specific P2 receptor antagonist. So this tells us that the presence of ATP in the tumor microenvironment helps in increased migration and invasion, or affords these metastatic properties to the cancer cell lines. Now, to look at what type of P2 receptors are present, again, we tested a few of these, not all of them. And among them, for example, HeLa cells strongly express P2Y6 receptors. Nearly all are expressed in all the cell lines, not that any is. So that's, again, the hurdle as far as a specific antagonist is concerned. But we found that in the case of IMR32, that is a neuroblastoma cells, and in the breast cancer cells, P2X7 antagonists were very effective as far as inhibiting the migration or invasion is concerned. So here we can see ATP increases the migratory capacity on both cells, which we can reduce when we use a specific inhibitor called A740004. The migratory capacity is also increased with PZ-ATP, which is a specific agonist and also reduced by specific antagonist. Same is the case with invasion assay. In the case of HeLa cells, although they strongly express P2Y6 receptors, when we looked at the receptor expression with respect to time after stimulating the cells with ATP, there is not much change as far as P2Y6 levels are concerned. They are more or less same throughout. But when we look at the P2Y other receptors, we saw a strikingly strong increased expression of P2Y12 receptor. Now, this suggested that probably when there is ATP in the microenvironment, then this particular receptor is getting strongly expressed, which may be the case in the case of chemoresistant cells also. So we tested it with a specific agonist, that is ADP. ADP shows a strong increase in the migrated cells. And we used a specific antagonist, ARC 69931, which reduce the migratory capacity in both ATP-treated cells as well as in ARC-treated cells. It's true for both migration as well as invasion. So this tells us that P2Y12 receptors are probably playing a major role as far as HeLa cells are concerned. Now, this is what we had seen is that the P2 receptors are involved. We just wanted to know whether downstream of the P2 receptors, COX2 is involved or not. And so for that, we treated the cells with ATP, and then we treated with specific COX2 inhibitors, indomethacin and celecoxib. And both of these inhibitors reduce the migratory and invasion properties in cells which have been treated with ATP. In all the three cell lines, we see that this property has been reduced, which suggested that ATP increases both migration and invasion in these cells in all the three cell lines, and COX2 inhibitors can actually reduce the effect. So that means the pathway that is followed is ATP is binding to the cholinergic receptors, and downstream COX2 is one of the enzyme that is activated, which we also found true in both the neuronal cells as well as in the cervical cell line. You can see that with ATP, and here there is a strong increase in the COX2 expression. And the P2X7 agonist, BZ-ATP, also increases. Same in the case of HeLa cells. The P2Y12 agonist, ADP, increases the levels of COX2. And the specific inhibitors, A74 for P2X7 and ARC for P2Y12, reduce the amount of COX2 or levels of COX2 expression in these cells. We also did digestion assay, which is a method to test the activity of matrix metalloproteases. And one of them, MMP2, is involved in it gives the property of invasion for the cells. So since it has to digest the matricial for the invasion properties, so we tested the activity of MMP2 directly in in-gel assay. And we can see that ATP increases. So the strong blue one essentially is where the digestion has occurred. So the gel digestion has occurred. So we can see a strong band in the cells which have been treated with ATP or the specific agonist, BZ-ATP. And again, it is reduced when cells are co-treated with the specific antagonist. And this is same in HeLa cells also. With the P2Y12 antagonist, it is significantly reduced. We then moved on from cell lines. We then wanted to check whether in animal model also this is true or not. So for this, we used C57 black 6 mice, which have been injected with a syngeneic cells. So we used EL4 lymphoma cells, which are syngeneic to C57 mice. So we injected them within in the shank area subcutaneously. So on day 1, and then after 10 days, a palpable tumor can be seen. When this tumor is detected which is a subcutaneous tumor, when it is detected, we injected the specific, we injected suramin here. In this case, we injected suramin directly within the tumor. And then we measured the tumor volume until for the next 1 week. And as you can see from day 10, where the tumor is palpable, the mice which have been treated with saline, there is an increase in the volume of the tumor size grows, and after a certain stage we need to euthanize the animals since the tumor size becomes too big. In contrast, in the mice which received suramin within the tumor site, where the volume strongly reduced, significantly reduced. So it's this shows that inhibiting the P2 receptors has an effect on the tumor volume, as we can see in the Hithany staining pictures here. And downstream, in order to see whether this effect is coming from the action of COX2, we can see there is a strong expression of COX2 in saline tumors, whereas in the tumors that are collected with suramin-treated tumors, there is a lower expression of COX2. And the same is true for the receptors. Both Y12 and X7 are reduced in these animals in the tumor. So this shows us that our hypothesis that when we treat the cells with when the tumor cells are treated with chemotherapy, for example, where here we have used doxorubicin, it causes increased cell death, which leads to the release of ATP in the tumor microenvironment. We hypothesize that this ATP acts on P2 receptors, which are present on cells which are now surviving, the drug-resistant cells, and that this will afford the migration and invasion property to these cells, as they are causing the metastatic properties. And we can block this metastasis by using inhibitors for P2 receptors. Currently, we are actually looking whether in the population of cells which are dying, whether there is specific what type of P2 receptors are expressed, and what exactly are these drug-resistant cells, whether there are cancer stem cells or other type of cell types, how this modification happens, or why they are in fact resistant. Are they resistant because they already express P2 receptors, or they express P2 receptors after the ATP is present within the tumor microenvironment? So these are the studies that we are currently working in the lab. One more question that we would want to then complete the talk with is how this exogenous ATP is involved in increased COX2 synthesis. So for this, we looked at some of the downstream signals, downstream of the P2 receptors. So in all these cells, we see that there is inhibitors of the MAP kinases. They reduce the levels of COX2. And so both the P38 MAP kinase as well as P42 MAP kinase, both of these are induced. So both of these are downstream of the purinergic receptors. And both of these, both P38 and P42, have already been known as a signaling towards a pathway towards increased COX2 synthesis in all cell types. What we checked for was the levels of COX2 mRNA within these cells. As shown here, this is in the macrophage cells again. If we treat the cells with LPS, there is nearly a constant expression of LPS. So there is not much fold change within the cells, whether you collect the mRNA at 4 hours, 12 hours, or 36 hours, up to 36 hours. The amount of COX2 mRNA remains more or less constant. Whereas in cells which have been treated with both LPS and ATP, we see that there is a strong increase in the expression of, or a strong increase in the amount of COX2 mRNA. So at 36 hours, the amount of COX2 mRNA in cells which have been co-treated with both LPS and ATP is almost 50-fold above four-fold above what it is seen in the LPS treated cells alone. And we know that there is, we also did this experiment with actinomycin just to check that whether there is increased synthesis going on or not. Now the question is why we have such high levels of COX2 mRNA within the cell. And we found that one of the reasons for this increased presence of COX2 mRNA within the cell is because there is a continued or sustained phosphorylation of one of the important kinases, which is the CDK9. This CDK9 is involved in the early transcription, and it is responsible for the phosphorylation of the serine 5 on the C-terminal domain of the RNA pole 2. So once this RNA pole 2 undergoes both serine 2 and serine 5 phosphorylation, that is when the RNA synthesis moves from the initiation phase to the transcription elongation phase. So this is very important for converting from the pause, from moving the pausing step in the RNA polymerase. So this is where the CDK9 works. In normal cells which have been treated with LPS, at 30 minutes, of course, there is a strong increase in phosphorylated levels of CDK9, so suggesting there is increased RNA polymerase activity. But in LPS treated cells, this activity reduces after 24 hours. There is nearly no expression here, but which remains sustained in the case of cells which have been treated with ATP. There is still a consistent amount of phosphorylated CDK9 even after 24 hours. So this suggests that there is increased activity of RNA pole 2 leading to increased COX2 synthesis even after 24 hours. Another example, another method, another pathway we also found is at the stability step of the COX-2 mRNA. If you look at the COX-2 mRNA, there are multiple of these shock domain sequences, the AU-rich sequences towards 3' UTR. And this 3' UTR determines the stability of the mRNA. And one of the ways in which this stability is mediated is through the activation of P38 MAP kinase, and downstream of the MAP kinase pathway, there is MAPCAP, K2, and other proteins which are downstream, which eventually have activate proteins and bind to the AU-rich sequences in the 3' UTR. Again, we found that in cells which have been treated with LPS and ATP, even after 24 hours, there is a significant amount of protein P38 MAP kinase that is still present within the cells. So this says that there is a prolonged or sustained activation of both CDK9 and P38 MAP kinase in cells which are treated with both LPS and ATP. And together, they ensure that there is increased transcription as well as stabilization of the mRNA. Also, proteins that are involved negatively cause splicing of the COX-2 mRNA, their levels are reduced in cells which are treated with LPS and ATP. So this, which is mediated by the HNRNP A0. So this is what we had hypothesized and published.
This meeting is being recorded.
Thank you. Can I take any questions?
Are there any questions?
Yes, sir. Just a second. We had some connectivity issue. We are just joining back.
Yes, I have a question actually. So, Professor Ravi, did you try replacing ATP with GTP or CTP and to check if they give the same result? Or is it very specific to ATP only?
ATP or CTP because the purogenic receptors are specific for ATP. ATP and ADP and for the adenosine. So these purogenic receptors are specific for these. So we tested only these.
Okay.
They do not bind with CTP or GTP.
Okay, they are highly specific. Okay. And they are activated because of this binding only, or there is any other mechanism by which these receptors can be activated?
These receptors are specific for ATP. And once ATP binds, that is when they are activated. And these purogenic receptors have multiple roles as far as the cell type is concerned. For example, as I told you, P2X4 receptors expressing on macrophages increases their activation and causes increased phagocytic activity. P2X7, for example, are also involved in neuronal cells where they cause increased calcium uptake because P2X7 is associated with an ion channel and thereby increased cytokine release. IL-1 beta release, for example, requires inflammasome formation. It requires a P2X7 receptor. So these receptors have multiple roles in physiology. The P2Y receptors are all G protein coupled. So some are GS coupled, some are GI coupled, and accordingly the signaling pathway manifests.
Okay. I would just like to commend your work. It was very meticulous. I'm sure even the undergraduate students who were listening, they will be able to get how you are delineating things step by step. Really interesting.
Thanks. Thank you.
Sir, can I ask one question?
Yes, ma'am.
Sir, like you have said that there is prolonged inflammation in the neurodegenerative diseases. Is there any cause or is there any particular reason why it is so prolonged? Because generally inflammation is helpful also.
Yes, inflammation is helpful, but as I said, inflammation, the third component of inflammation is the resolution of inflammation. And this resolution has to happen so that the tissue repair can actually happen. And resolution can be done through various methods. There are various ways in which resolution happens, including chemical mediators, which are known as resolvents. So this is our next topic that we are going to work on, whether the expression of these resolvents are reduced for example, in neurodegenerative conditions or other cases of inflammation, whether there is a change in the levels of resolvents, or why the resolvents are not capable of doing the repair mechanism. And also, as I've said, while PGE2 is responsible for many of these inflammatory conditions, there are other prostaglandins which are also synthesized. We need to also check whether PGD2 levels, PGF2 levels, these are changed or not, because these are actually anti-inflammatory and they help in the normal process of inflammation, they come in later on. So in the beginning stages, there is an increased activity of prostaglandin E synthase, and there is increased PGE2 levels. And eventually there is an increased synthesis of PGD2 and PGF2, so which come later on. So whether that can be why that process happens late, or whether that doesn't happen in chronic inflammation. So these are some things we need to check.
So there's one more question, like P2 inhibitors, they are involved in they're acting as anti-inflammatory. Can they be used in the COVID-like situations also where inflammation occurs?
Hypothetically, yes, but we have not tested it. So we had checked it only in different cell line models, in monocytes, macrophages, and microglia. We have checked with LPS as a stimulator only. We have not checked with other kinds of stimulants which can also increase inflammation, like beta amyloids or any viral proteins, for example. So these we have not checked. We have done in tumor cells. Again, there we have checked, but we have not done other stimulators for inflammation. But in theory, we think that it should be valid.
Thank you so much, Professor Akundi, for such an insightful lecture. And the topic was really relevant to the current context. Thank you so much. Thank you.
Thank you.
We'll now take a short tea break. I request all participants to please rejoin by 3:30 p.m. for the third and the last session in which Professor Ajay Paul and Professor Abdul Haq Ansari will share their valuable insights with us. So see you at 3:30 p.m. Thank you.
The link for feedback and certificate will be shared in the Zoom meeting only. So remain joined till the end to get the certificate.
Welcome back to the final session of the IBRO-APRC symposium. I hope everybody is back to the session. So the first presenter in this session will be Professor Ajay Pal, who is an associate scientist at Shriners Hospital's Pediatric Research Center for Neural Repair and Rehabilitation at Temple University School of Medicine, Philadelphia in the United States of America. Dr. Pal is also the guest editor for Brain Science special issue on brain and spinal cord electrical stimulation and executive editor of International Journal of Medical Laboratory Research. Professor Pal's presentation on spinal cord associative plasticity improves forelimb sensory motor function after cervical spinal cord injury will focus on the application of paired stimulations to enhance sensory motor recovery in cases of spinal cord injury. Professor Pal, it's a pleasure to have you with us today, sir. We look forward to an interesting and engaging session on this interesting topic. Thank you.
Professor Pal, your co-host.
Thank you for. Okay. Can you unmute my video so that I can start video?
Yes, you have the right. You are co-host now. You can.
Okay.
Present.
Can you all see my presentation and hear me properly?
Yes, sir. Yes, sir.
Okay. Good afternoon, everyone. Now it's good morning for me. But and I thank Dr. Suri for inviting me for this wonderful event. And I look forward to have a wonderful discussion on my project and the work. And the title of my talk is Spinal Cord Associated Plasticity to Improve Forelimb Sensory Motor Function After Cervical and Spinal Cord Injury. So for the today's presentation, I'm going to discuss upon the sensory motor integration, especially targeting the spinal cord at the site of sensory motor integration. What happens to the sensory motor integration after cervical spinal cord injury or spinal cord injury as a whole? And how we think the associative plasticity can improve motor function or recover motor function after injury. Then the second part of my talk is focus on my work, how we think the spinal cord associative plasticity result motor function recovery after spinal cord injury, and our trial of this spinal cord associative plasticity to recover motor function after cervical injury. Sensory motor integration is the basic for any skilled movement or locomotor function. And in central nervous system, we have multiple the skillful movement like primary motor cortex, cerebellum, parietal cortex, basal ganglia, spinal cord, brainstem. So my talk is focused on spinal cord as a center for sensory motor integration and how we can recruit sensory motor learning in the spinal cord. So what happened after cervical spinal cord injury or any spinal cord injury? It disrupts the sensory motor integrity. What happened like in this picture, so that after impact to vertebral body or any jerk of forceful impact to bone, vertebral bone, it herniate and damage the spinal cord. And due to this impact or the acute injury, there would be a differential loss of sensory motor function dependent on the extent and level of injury, loss of autonomic function, bowel and bladder control loss, and it develop a consistent pain. And problem is there is no cure for this kind of injury. So we are still looking for a proper cure or proper management of this spinal cord injury. And what happens after injury, it has been found that corticospinal system, especially the descending corticospinal system, and the impairment or degree of impairment is closely related or linearly related. So if you damage more severe or severe spinal cord injury that results the severe corticospinal tract injury, it results a severe impairment. I can say in another word, to recover the movement or recovery of motor function, we need to fix this corticospinal system. So associative plasticity, when I talk about the associative plasticity, the first thing come about in my mind is this conditioning experiment by Pavlov, or it's like simple conditioning experiment where you pair the two stimuli, and depending on the timing of these stimuli, you can induce a plasticity in the system or the learning in the system. Here, food is the unconditioned stimuli, and but it paired with a conditioned stimuli, just ringing the bell. And when they are properly timed, even when you remove unconditional stimuli, you can induce plasticity or induce a response. And when they are repeatedly paired, you can induce the learning in the system. So using this associative plasticity principle, we can also induce plasticity in sensory motor system and get some functional recovery after injury. So I'm going to show some study which target sensory motor integration for movement recovery. First classical example is this pre and post stimulation that we also call Havian plasticity. So what happened when you stimulate a presynaptic input followed by the postsynaptic input, and when you do repeatedly, you can induce a postsynaptic plasticity in the system. And this can lead to a functional recovery to some extent, and people has tested. Then second is the way we are doing, like my whole talk is going to based on this sensory motor integration, where we think descending motor system and sensory event, when they meet in the spinal cord, and if we repeatedly make them to stimulate them, we can induce plasticity in the system, and that can strengthen the spare circuit and get the motor recovery after injury. The third sensory motor integration protocol is to stimulate motor cortex, the phasic stimulation of motor cortex and tonic stimulation of spinal cord. And when you do repeatedly, you can induce plasticity in the sensory motor system, and that can recover the loss of motor function after it. So what is the important component of this type of plasticity? It integrates both sensory and motor system, and it need at least some spared connections to be strengthened by repetitive stimulation or to make it associative when we do repeatedly to induce plasticity in the system. So in our spinal cord associative plasticity, and why we call it as a spinal cord associative plasticity, because we are integrating descending motor system, whichever is left after injury, and intact sensory system, here the sensory afferent, in the spinal cord. When we repeatedly stimulate brain and spinal cord, we can induce plasticity and strengthen those spared connection, and that lead to functional recovery after injury. Critical component here is the sensory motor integration in the spinal cord, and we need at least a spared connection to get it integrated. And why we think is can be translated to the clinic, because people are walking with a spinal stimulator. So spinal stimulator for pain management is already in the use. We can use a spinal cord stimulation with the spinal stimulator, and RTMS stimulation or transcranial magnetic stimulator to the motor cortex, and induce this kind of sensory motor integration in the spinal cord. and we think it can result sensory recovery after injury. And our long-term aim is to get motor function recovery after spinal cord injury. And we hypothesize that this associative learning mechanism can be induced in the spinal cord with paired electrical stimulation of this paired synapses of motor descending motor system and sensory apparatus. So how we do this spinal cord associative plasticity mechanism and how we target it. So first we are doing all the experiment in awake and behaving animal with implantable electrodes. So I'm going to talk about here how we did our experiment. So to stimulate descending motor system, we stimulate motor cortex with 3 pulses at 333 Hertz. Then sometime later, we stimulate spinal cord at single pulse. And here our motor cortex stimulation is supra-threshold, while our spinal cord stimulation is sub-threshold. So supra means like if you stimulate with supra-threshold current, you can induce a muscle twitch or EMG that we call. But sub-threshold stimulation, it doesn't cause any muscle twitch. So, and we measure our evoked responses from bicep muscle with implanted electrodes. To stimulate spinal cord in awake animal is challenging because it's like we are doing cervical injury and we have to stimulate the rat neck, which is quite movable when rat is awake. To do so, we collaborated with biomaterial scientists in UT Dallas, and we developed a spinal cord stimulator. Here I'm showing the 2 channel of the stimulation. And the property of these electrodes, when at the time of insertion, like at the room temperature, they are stiff. But when you place them inside the spinal canal, right? I did the laminectomy as C4, then I slide these devices in the spinal canal epidurally. So with change in temperature from room temperature to physiological temperature, it makes these polymers supple and it confirms the shape of the spinal cord. And that we observe like after implantation and postmortem imaging, this MRI imaging, and we see at the time when it is flat, and then after some time later it get softened and take the epidural surface. So with this system, we can stimulate spinal cord without causing any damage to it. The next thing is like, as I said earlier, the timing is a critical component for associative learning. Now these experiments, which I'm going to show, they focus on the critical timing for the sensory motor integration in the spinal cord and why we think that particular timing is important for associative plasticity and to result learning. To assess this, we implanted motor cortex electrode here. For the baseline measurement of physiology, we stimulate motor cortex at 110% of cortical threshold. So threshold is the least amount of current needed to induce a muscle twitch or EMG response in more than 50% of trial. And to pair it with the spinal stimulation, we are using sub-threshold, that is 90% of spinal threshold. So at the threshold, with the spinal stimulation, you can induce a muscle twitch with the EMG in the bicep. But at 90% of the spinal threshold, we cannot induce any muscle twitch or EMG. So idea is like sub-threshold spinal stimulation is not causing any change to the spinal system or not inducing a muscle response, but it only potentiates the descending signal which is coming from cortex stimulation. And when we pair these stimuli at different time interval, so in this picture, I'm quantifying it, the 0, any change to the y-axis percentage, 0 is the only motor cortex stimulation response. And whatever positive to that is with addition of spinal stimulation. And the simultaneous stimulation at 0 in the x-axis shows when these motor cortex and spinal cord stimuli are delivered simultaneously with no delay. So negative axis is showing when we first stimulate the spinal cord followed by brain or motor cortex, and the positive time axis is showing when we stimulate first motor cortex followed by spinal cord stimulation. And this graph is belong from the intact animal with no injury. So here, as you can see, when we stimulate motor cortex first, at from 5 to 15 milliseconds later, spinal cord, we see a stronger motor evoke responses compared to the baseline, which is only cortex stimulation at 10 milliseconds after brain stimulation to the spinal cord stimulation. And the negative act is when we stimulate spinal cord first and then sometime later brain stimulation or motor cortex stimulation, we see some kind of augmented responses and significant at the time is -20. That means spinal cord stimulation followed by brain stimulation in delay of 20 milliseconds. So from these experiments, we get there is a augmentation in motor evoke responses or descending motor evoke responses with paired spinal stimulation when they are properly timed, and here is the 10 millisecond latency is the optimum. Similar thing we observed when we repeated these experiments in animal with spinal cord injury. Here also we get this 10 millisecond latency from brain to spinal cord stimulation is critical and in augment a larger increase in motor evoke response. So why this 10 millisecond latency is important? So to know that, first we stimulated the brain of motor cortex and recorded the dorsal potential from the spinal cord epidural surface from C5, C6. And we see the latency from brain to spinal cord responses that comes around. So this time is critical when we pairing spinal cord stimuli with brain after 10 milliseconds, that is very close to 9.5 milliseconds. Then it potentiates the descending motor evoke potential. So the timing of 10 milliseconds that we get it from here, we use that 10 millisecond pairing in our further experiment to induce plasticity. Then the question is like, after stimulating spinal cord, are we recruiting sensory afferent or this proprioceptional afferent system? To know that, we did loss of function studies, and also first we tested this like the property of afferent recruitment with the spinal stimulation. So here in this experiment, in acute experiment with anesthetized animal, we stimulated spinal cord, dorsal side of the spinal cord, dorsal root entry zone, this is the blue, where the afferent enters the dorsal spinal cord, and the dorsal root ganglia. So we see that in these three stimulation sites, we did not see any difference in latency of these responses that is recorded from bicep muscle. So these three different sites, they follow similar kind of response. And when we repeatedly stimulated with 100 Hertz, so there's a property of afferent stimulation when it's called the post activation refresh. So what happens when you stimulate sensory afferent with like continuous stimulation or like high frequency, here I'm using 100 Hertz stimulation, delivering 15 pulses. So after first stimulation, you will see the augmentation of its response to the second one, but adding subsequent stimulation, it suppresses the responses. And that we observed from all three sites of stimulation. So from these experiments, we get that if we stimulate even the middle of the spinal cord, we are able to recruit sensory afferent with the spinal cord stimulation. So then we tested like this, we are saying sensory motor integration in the spinal cord, and does this descending CST or sensory afferent are important component for dissociative plasticity. to test that, we inactivated descending corticospinal system and observed the effect in physiology and function. To do that, we use a DREAD system. It's like designer receptor executive activated by designer. So what happens in this system, you can modify a receptor, and so that that can be activated or suppressed only with a designer drug. In this case is clozapine and oxide. So we use a 2-virus system here. We injected DREAD in the motor cortex and a tree-dependent virus in the spinal cord. The idea is when these doubly infected tract can be inactivated with this external lab here in the CNO. And we see the expression of this DREAD in the motor cortex, lab 5. So, and we tested the behavior before CNO injection. So for the behavior measurement, we give animal, here in this case is a rat, eat pastas like uncooked pasta, and the way it manipulated fingers while eating this pasta, piece of pasta, that we are calculating this like percentage of paw adjustment, both left and right. The baseline is like whatever we get before inactivation, before CNO injection is 100%. So here is like we injected contralateral, we are inactivating only contralateral surface. So one side of one paw is impaired and another paw is not impaired after CNO. So for the inactivated side, the number of manipulation is decreased to around like 40% to the before inactivation after CNO injection, and that is 30 minutes after when effect of CNO is peak. And the non-inactivated side, the other paw has no change in number of manipulation or the paw aspect, and it recovered back after 2-hour washout period. Similarly, we measure the physiology during activation, and here I'm measuring that like time experiment. What I saw, the paired stimulation of brain and spinal cord. The baseline is whatever pairing effect we get before CNO injection, and the time points after CNO injection. And you can see it's like at the around 30 to 40 milliseconds, this effect of pairing is completely washed out. So this CST is now completely inactivated and it has minimum effect in the other half. So from these experiments, we know that this descending corticospinal system is an important component for this associative plasticity. Now, second is the sensory afferent. So work out that the necessity of sensory afferent for the spinal cord associative plasticity. To do that, use two different kind of system. First, we use the simple DREAD system. Here we injected this DREAD virus that I saw in this slide. This is like DREAD in the dorsal root ganglia, and we measured physiology before CNO injection and after injection. You can see the expression of this DREAD level DRG neurons. And after inactivation, we silence the sensory afferent. We are targeting here mechanoreceptor and proprioceptor, and that happens around 30 to 40 minutes after the drug injection, and it affects minimum to the other part. The second strategy we used specifically targeting proprioceptive afferent, because this like proprioceptive afferent that keeps a position of your limb in space, like, and muscle tension and length, the information of length and tension to the brain. So we inactivated using this drug, this proprioceptive afferent in a PV mice. So if these are PV is like palpebral mice. So they have to their palpebral. So palpebral is up exclusively expressed in the proprioceptor in the DRG. So when we target these proprioceptor, we can inactivate with the inhibitory drive, and we get a similar kind of effect here also. After inactivation with CNO, there's a loss of paired response to brain and spinal cord stimulation. So from this experiment, we know that the afferents are necessary for effective corticospinal pairing. So now we develop a protocol to induce plasticity. So before these experiments, we are just testing a pair of stimuli, like single stimulation to the brain followed by spinal cord stimulation. But this single pair of stimulation plasticity in the system. To induce a plasticity, the stimuli should be repeatedly presented. So to induce the plasticity in sensory motor system, first we measure a baseline response either by motor cortex stimulation or spinal cord stimulation and their threshold. Then we repeatedly pair this motor cortex and spinal cord stimulation at 10 millisecond interval. With this repeated pair stimulation protocol. So like, is it that first we deliver 3 pulses in motor cortex, 10 millisecond later a single pulse at the spinal cord, and these are delivered at 5 Hz frequency and for like 150 pairs in 30 seconds. After 30-second pairing, we give us 1 minute pause, then repeat this pairing and pause for 20 times. So total time duration is 30 minutes, and we pair a repetition of cortex and spinal stimulation 3,000 times. And we think this 30-minute time is critical because it's like to even the electrical stimulation to the humans or the pairing stimulation. They say 30-minute time windows important because 30-minute time window can be tolerable for the patient. You cannot hold a patient to or the subject for than 30 minutes or hour for a stimulation period. So we kept the timing for 30 minutes. Now we think this 30-minute protocol can induce plasticity in the system. So in a single session of 30-minute protocol, that has a lasting effect in the system. So here I'm showing the percentage increase in MEP after 30 minutes of that repetitive paired stimulation. And the y-axis is time after repetitive paired stimulation. Here we are measuring only baseline response, either cortical or spinal cord response. So you see, just after 30-minute pairing, you see the enhanced motor evoke responses, and that happens to the black and this gray line is belong to animal, uninjured animal, and the red lines are like responses from spinal cord injured animal. So both uninjured animal and spinal cord injured animal, there's a lasting change in motor evoke responses just after 30 minutes of repetitive pairing. So we think this repetitive stimulation can induce plasticity in the system. And because it recruiting the descending corticospinal system which are spared after injury, and we have the spared sensory afferent, and we are integrating that repeatedly in the spinal cord to induce plasticity. To use a spinal cord associated plasticity for moment recovery after injury, we have this timeline of segment. So first we measure the S reflex responses that I will explain later before injury and their baseline behavior of the animal. Then we do spinal cord injury. Here I'm doing C4 contusion injury, like 200 kilodalton contusion injury, a moderate spinal cord injury. Then we give 1 to 10 days of rest, where we just measure the baseline response, like H reflex and behavior. Then we stimulate animal starting from 11th day of injury to 20th day. We are giving 10 days of 30-minute session of stimulation each day. And during that time, at some point, we test the physiology, like I measured the immediate effect of pairing. Then every 2 weeks after stopping this paired repetitive stimulation or SCAP protocol, we measure their behavior and physiology. At the end of 7 weeks, or like we design it to for only 7 weeks or 50 days after spinal cord injury, because in other similar kind of paradigm, we observe that there is an enhanced motor function recovery after 50 days of injury. So primary outcome measure here is like behavior measurement of IBD, that called like finger manipulation or dexterity. And after 10 days of stimulation, when of repetitive pair stimulation or SCAP protocol, we see it induces plasticity in terms of like enhancing the motor evoked responses with paired stimulation compared to the controlled element which does not receive any SCAP. So what about 11th day, they have similar augmentation in motor evoked responses. At day 15, like 5 days after the repetitive stimulation, there's a significant increase from the control animal, and that happens also increase further at the end of 10 days of stimulation. And after stopping stimulation, it has persistent increase in motor evoked responses compared to the control. So from this, we conclude that this SCAP of paired stimulation can induce plasticity in the system, and it has a cumulative effect with added day of stimulation. So one of the important thing is like spinal cord injury induce spasticity or hyperreflexia after in humans or also in the animal. So to measure the hyperreflexia, we tested H reflex modulation. So what is H reflex? So H reflex is a kind of knee jerk reflex. So it's like spinal simple reflex, spinal circuitry reflex. So what happens when you stimulate a peripheral nerve, you can stimulate directly and record it from an distal muscle. So you will get a direct muscle evoked response that is called MWave, like EMG. And the second, you will get sometime later this spinal circuitry or spinal loop response that we call the H reflex, because the scientist who discovered the Hoffman who discovered this reflexes. So here for measure H reflex, we stimulate ulnar nerve at the wrist with a pair of wire electrode and record EMG responses from abductor digiti minimi, the tiny muscle in the wire. And when you stimulate a nerve, you will see a direct muscle evoked response and sometime later H reflex. So H reflex comes first, followed by at low intensity, and then when you increase the intensity, you will get the M response. And when these pulses are paired, like here, when I deliver a pair of pulse, you can modulate the second response of H, but it not change the motor evoked response or M wave. And that depends on the frequency or interstimulus interval. So what happens when before injury, when we test a different time interval between first and second stimuli, and we measure the second percentage change in second H response compared to the first one, in the normal animal, both stimulation and control, we see this kind of frequency dependent suppression of H response, the second. After spinal cord injury, this frequency dependent suppression is abolished, and that happens in both control and stimulation. After 10 days of repetitive stimulation or 10 days of SCAP, we see this modulation is restored in animals that receive stimulation. Associated plasticity can modulate a spinal reflex system in a positive way and made it towards the normal. Then we measure our behavior test. So for behavior measurement, we use a dexterity score that called IBB, like Irvine, Besso, and Betty. These are the scales. So what they do, you rate a finger movement or foot manipulation, score it in a way that how much finger or grip or this is involved. So maximum score in normal condition is 9. After injury, it decreased. Like here is like when we measured second time. So the score 9 is to like 2.5. And with 10 days of stimulation, you can see in animals that receive stimulation, there is significant recovery in movement or dexterity compared to the control animal. So as I said, like we timed it to 50 days after injury only, and because our primary outcome measure was this as you can see, the animals which received stimulation, they have not significant, but a trend in decrease in error on the ladder walk compared to the control animal. So these 10 days of SCAP can induce plasticity and recovery after spinal cord injury. So the question happens, what happens when you induce a contusion? Does it cause a similar injury to both group of animal or there is a difference? So then we analyze the contusion curve. So here we deliver 200 kilo force to the spinal cord at C4 level to injure. And these are the different animals which receive contusion, and we see there is no difference in injury just because of the impact. And this is example of one of the injury, how it looks at epicenter and rostral and caudal to the epicenter. Then we quantified spared tissue and spared axons. Spinal cord starting from C3 and C7. Injury center, like the site where injured is C4, so the segment above and the below segment. And the red here is shows the area which is completely damaged. And these are the like purple ones are the spare tissue. And when we compare the spare tissue area in both group of animals, we did not see any difference. So the tissue sparing is similar. Similarly, when we quantified motor and sensory axon length below the injury, we did not see a difference in across the two groups. This we say that like injury, there's no difference in tissue sparing, and there is no difference in axon length. Means the plasticity which is happening is just not making a specific connection. Maybe I'm just speculating that, but it's just strengthening those systems which are spared. So in conclusion, this spinal cord associated plasticity, which we recruit by stimulating descending motor system and sensory afferent and doing repeatedly, it can induce plasticity in the after spinal cord injury, and it recovered the loss of motor function. And we saw that here both corticospinal system and proprioceptive afferent are necessary for this kind of plasticity. And there are spared circuit which can be targeted with this plasticity to enable recovery of function. And this 10 days of stimulation, it improve physiology and behavior, also it's improved the reflex modulation. I didn't do all the experiment, it is a team effort. So, and there are people who involved in different set of experiments. I want to thank them. So initially we started this work in Burke Neurological Institute, then our lab moved to the Columbia Medical Center. And most of the work we did in the Columbia. So Jason Carmel is my PI, was the lab head. And Hong, Aditya, Ahmad, Thelma, and Meenu, they are involved in, like, I mostly did the physiology part of experiment and surgery. Hong was involved in inactivation experiment. Aditya and Thelma, they were involved in behavior experiment. Meenu did some of the histology, and Ahmed also did some pairing experiment. And we also thank our collaborator from UT Dallas, Walter Wyatt, to develop the spinal stimulator for us and our funding agency. And thank you all so much. I welcome any questions.
Thank you very much, Professor Paul, for such an enlightening lecture. So there are some few questions. This paired stimulation, can it be used for the recovery after stroke?
Yes, definitely. Even in our lab, we have this subcortical stroke model where we are using it. So it also induces plasticity.
And how long the effect would be there of this paired stimulation? And what should be the frequency of this treatment?
Yeah. So if you see, it's like I designed it for only 10 days of stimulation. And it has a cumulative effect. And if you see this slide, so it has like after 10 days, at day 20, we stop stimulation, but still we see potentiation in motor nerve responses. So it's like this therapy is what we are designing, or what we are hoping with a certain period of stimulation, it can retain the engagement in the system. So you do not come for a repetitive stimulation at some interval time. That's our hope. It's not like prosthetic kind of thing, but it's like permanent changes in the system.
So like these experiments have been done in the animals. Have they been tried on the human also, or it is still on the animals?
So not completely, but we have a, like now I mean the Jason, they are doing and this paired stimulation in patient with spinal stenosis. So it's like you have the injury to the, especially cervical spinal stenosis. And luckily we observed a similar kind of potentiation when we paired those stimuli. So idea is like to engage descending motor system with this repetitive transcranial magnetic stimulation and to stimulate spinal cord with the implantable electrodes. So there are implantable electrodes and people are walking with those implanted stimulators. So that give us an opportunity to use both systems, and it's not very chronic, like you can stimulate it and pair these stimuli. So if you are fundamentally correct, we can definitely move it to clinic.
So in future, we can see this can be used.
Yes, definitely. That's our plan. Yeah.
Thank you so much, Dr. Paul. Once again, it was really an enlightening lecture. Thank you so much. Over to Imran.
Thank you, Dr. Paul. Our last speaker today, Professor Abdul Haq Ansari, will be focusing on a very exciting development in neuroscientific research. Professor Ansari is a faculty member at the Department of Physiology, Nepalganj Medical College in Chisapani, Banke, Nepal. He is also the founder and executive member of Nepal Neuroscience Society. Professor Ansari had organized the IBRO-APRC Associate School in Nepal in 2021. His presentation today on use of transcranial magnetic stimulation in neurosciences will introduce us to repetitive transcranial magnetic stimulation, a new neuromodulatory technique in neuroscience. This technique is a non-invasive brain stimulation management strategy used to treat many neurological disorders and has great potential for the future. Professor Ansari, we have been eagerly waiting to hear from you, sir.
Professor Ansari, your co-host now. You can share your screen.
Thank you. Hello. Good afternoon to everyone.
Good afternoon.
Good afternoon, Dr. Abdul. Welcome.
Good afternoon, ma'am. Okay. I think I'm audible now clearly. Hello.
Yes, yes,
Hello.
Yes,
slides are
Dr.
visible. Your slides are visible.
Thank you. A special thanks to organizers for giving me this opportunity to talk. And the symposium, the topic is use of repeated transcranial, sorry. The symposium talk very exciting, that is recent trends in brain research, unlocking the mysteries. I was due to busy schedule. I just attended few, and last lecture I was able to attend of Dr. Ajay, very exciting lecture. So, and as I am the last speaker, everybody is tired, so I will try to finish it early. So the topic is use of repeated transcranial magnetic stimulation in neurosciences. So I will start with the simple, what is transcranial magnetic stimulation, introduction, then how it works, and why we say instead of transcranial magnetic stimulation, repeated, RTMS in short, then how many TMS treatment sessions are needed to show the efficacy, to show the effect, therapeutic effect, how long until the RTMS treatment starts working, then how long do the effects of TMS treatment last. Okay, and in the last, I will try to show my experiments. So when I was going through the literature, I saw that with the invention of, it was done long before, even before the development of electricity. Egyptians were doing the brain stimulation therapies from the sea animals. As we know, different sea animals having a special currents there. So it plays role in treating neurological and mental disorders. Especially, these therapies are given as an adjunct therapy. Brain stimulation therapies are given when the different diseases which is cannot be cured with the drugs or by the physical any other means. Then as a support, we use the brain stimulation therapy. It involves especially activating or inhibiting the brain directly with the electricity, or electricity directly we are putting the electrodes as discussed by the Dr. Ajay, different type of devices now implanted into the brain, and they are giving either direct or pulse type of different modes of the electricity. And another way is giving the electricity non-invasively through the electrodes placed on the scalp. And especially for the transcranial magnetic stimulation, they are using magnetic field. As we know, when we flow the electricity into the wind wire, then it will create the magnetic field and they will generate the electrical activity. Okay, electrical through the electrical field. So repetitive transcranial magnetic stimulation is working on this concept. It is a coil, having different type of coils, and very small duration and very high current is given, which will generate very high electrical field, and that will affect the our brain neuronal resting membrane potential. And this, so this type of TMS, first portable type of TMS, is discovered in 1985 by three discoverers, Zelinus, Freestone, and Tony Barker. And they are especially mostly used for the different type of psychological and neurological disorders. As I told, a special type of disorders which is not treated by the drugs. Drug resistant type of diseases are treated by this repetitive transcranial magnetic stimulation, like depression, psychosis, anxiety, and many disorders are under the trial. It can be this repeated transcranial magnetic stimulation is targeted at a specific brain areas, like some studies show motor area, some show dorsal lateral prefrontal cortex. Okay, so different areas they are targeting, and very focal type, very pointed type of stimulation is done, and that is possible with this magnetic stimulator. So this is the device. It is a very simple one, and the leader of this manufacturer of this device is Megastim, and this is the Megastim device. And now if we see very sophisticated type, along with the MRI equipment, EEG, all are clubbed together, that type of new devices are now used in more sophisticated way. As this device contains one here central unit, and we can see here it is connected with here at the point B, which is removable. We can remove and we can use this. It is the electrode. So it contains electrode here, and this is the coil. This one, if you so you can follow me with the cursor. This coil is like H-shaped coil. Different type of coils are used according to our need. So this coil is H-shaped coil, and here we have to put this on the targeted brain, and it will generate the electrical field, and this will either excite or inhibit the area according to the frequency. And we have here knob, a very large knob. We can adjust it to increase or decrease the strength. Okay, amplify the power. So here it is showing 48, so we can go till 100. It starts from the very low, and we can, according to our need, we can increase the power. This figure is showing the electrical field generated after putting the coil, and this coil, when it is put here, it will generate, but this is generating the field, and hardly it is penetrating beyond 2 to 3 centimeter. Its depth is only 2 to 3 centimeter. Okay, it is not very strong. And this is showing different type of coils. Even this transcranial magnetic stimulation is under the trial, and different animal experimentations are done. So some coils are used specifically designed for the animals, different type of animals. Then different type of neurological and psychological disorders. According to that, different type of, but more popular type of coil is this H-shaped coil that is used in the most of the lab. Hello. And I have here copied some recent studies. Like this is showing LTP like cortical plasticity predicts conversion to dementia in patients with memory impairment. So they used for the to induce the cortical plasticity for the short time. This is used, as well as we can demonstrate this RTMS. We can demonstrate to show the nerve conduction pathway. What are the motor area in the brain? What are the sensory area in the brain? We can use in our labs also to demonstrate for the undergraduate medical students. Another, for the short term study in this paper, they have used Penn's adverse impact on training, induced performance and neuroplasticity. So for the short term, neuroplasticity they are using for the very short period of time, like 1 hour, 2 hours. Then they are checking the how much changes in the excitability of the neuron taking place for the very short type of experiments. This is showing effectiveness of high frequency left dorsolateral prefrontal cortex, RTMS, on depression, response inhibition, cognitive. So along with the depression and other diseases, for the cognitive test, okay, experimentally, we can show the cognitive impairment through the use of RTMS. Assessing the effectiveness of RTMS in treatment of obsessive compulsive disorders. So these are short type of experiments. In this study is very interesting, neuromodulation and eating disorder. So they are checking what are the changes, okay, in the eating disorder with the giving the RTMS. So these are showing the new trends, use of the new trend RTMS in other type of diseases like eating disorder and other things there. Now, how do you place this coil as a coil of the RTMS over the head? So for the different clinical setup, they are targeting different parts of the brain, and a specific part of the brain is excited or they are stimulated by the RTMS. So accordingly, it is most of the time, most of the clinical studies, they are targeting prefrontal dorsal cortex. And for that, first they start, okay, with the determine hot spot on the head that would induce a thumb twitch. So it is a by trial method, they are trying to find out, okay, appropriate area where when it is stimulated, they will induce a thumb twitch in the contralateral side. So if we are stimulating on the right motor side, it will induce the thumb twitch, okay, if it is reaching the beyond threshold level, then we are determining that that is the motor region, and that is called motor hot spot. So we have to find motor hot spot to find the appropriate area for the proper stimulation with the RTMS. As I told, most of the time, they are some of the studies mostly showing left dorsolateral prefrontal cortex. Some studies showing right dorsolateral prefrontal cortex. Both are having same type of efficacy. So we have to select either right side or left side motor hot spot. Then it is called 5 centimeter rule. So most of the time, after selecting the motor hot spot, we have to move forward anteriorly along a parasagittal line from the motor hot spot. How much? 5 centimeter. Okay, this is a very crude type. And to arrive at left or right dorsolateral prefrontal cortex. So this is showing the appropriate area. Okay, and then we have to here now in most of the devices, how much frequency we have to give and for what time, they are already we can set there. We can calibrate and we can put it there. Now, this is the very primitive type of device. As I told, I am holding the coil and putting there. But now, this type of automatic with a stand and everything there, okay, and very good reclining chair. The new devices are found, and we can set here how much time we need, how many frequency we have to give. Everything is automatically we can put and it will work fine. So another thing is how much strength we have to give. So as I told, first we have to start with the error and trial method. We have to start from the minimal strength, and will start. with a minimal strength, and we will go higher, and we will find the strength at which we will see the twitch in the thumb, in the contralateral side. So minimal strength which will induce this type of twitch in the left pollicis brevis muscle, that is in the left thumb, by stimulating on the right side. That is called resting motor threshold. So most of the studies, they are putting the strength of the stimulation from 80 to 90% of this motor threshold to 120% of the motor threshold for the very good result. So after selection of the optimal scalp area, we have to move on the anterior side, how much? 5 centimeter away. So that is the thing. And how much strength we have to give? As I told, 80% to 120% of the resting motor threshold. And when I use, it was almost 48%, means total strength, maximum strength which we can give through the RTMS is around 2 Tesla in most of the devices. So 50% of the 2 Tesla is sufficient to cause the twitch. How it is given? It is given in the pulses. So an electromagnetic coil is held against the brain, and it will give in the pulses. And we say hertz, like 1 hertz and 0.5 hertz, it is called low frequency. More than 5 hertz, it is called high frequency stimulation. Figure 8 coil is used. That is also called butterfly coil. And a typical RTMS session lasts from 30 minutes to 60 minutes. Okay, and they does not require anesthesia. Person is sitting comfortably, make person our patient comfortable, then put at appropriate place, select the threshold value, then adjust at 80 to 120% of the according to your study, and give in the pulses. So what is the mechanism of action? It is generating the magnetic pulses, and it passes easily through the skull, and it will create the small electrical currents, and this will expose, this electrical currents will expose the nerve cells. And as we know, we have potential, they have membrane, resting membrane potential, and this resting membrane potential is getting disturbed, either excitability or inhibitory effect there. So neuron membrane potential changes, as it will cause change in the conductivity. And when it reaches a certain threshold, that will fire action potential, and it will show the behavioral effects. So frequency of stimulation is high frequency, generally it is excitable, 5 hertz or more. Low RTMS, that is called low frequency, that is generally it is decreasing the excitability. So generally it is given in the pulses, like in my study, I choose low frequency, that is 0.5 hertz, and how much strength given was 120% of the motor threshold, and it was given on the right dorsolateral prefrontal cortex. And this green color showing 5 minutes, it is given at a rate of 0.5 hertz for 5 minutes, then 5-minute rest, then again, so 8 sessions, 5-minute interval, 40 minutes it is given. So what are the contraindications? What type of patients we don't have to take? Okay, anyone having brain damage or anything, so we are exciting the brain, so we have to exclude the patient having more brain problems there, or some device is already implanted there, like pacemakers or any medication which can be affected by the RTMS, or childbearing age, lactating women. This type of patients are not given the RTMS. Epileptic patient or seizure, because we are increasing the excitability, we are changing the excitability. So epileptic seizure can be induced with this RTMS. So that type of patients are not taken. As depth of the, it's how deep it is going is not more than 2 inches. Hardly it is going 2 to 3 centimeter deep from the skull. And it interfaces generally between gray matter and white matter. So from where that we say trigger zone, most of the trigger zone, and they will cause activation of the neurons. And its strength is maximum 2 Tesla, around 2 Tesla magnitude. This is already covered. What are the RTMS side effects? As it is very small area of the brain targeted, so it is more safer than the other direct stimulation of the brain. But mostly the reported RTMS side effects are, it will cause, in few patients, it will cause dizziness or lightheadedness. Okay, generally, very, very rarely, seizures are reported, but no death, suicide, etc. reported till now. No adverse effects on cognition till now. Most common side effects are headache, scalp discomfort, and as we giving above the threshold, so it will create the sound, okay, and it will like a scalp discomfort, very, very common, okay, reported side effect. Another thing is how many TMS treatment sessions are needed. So that also variable according to the severity of the patients. More severe patient, old patients, they require more sessions. Every day, it lasts from half an hour to 60 minutes. Low frequency, it will take more time. High frequency, it will take lesser time. Another thing is high frequency. Nowadays, mostly high frequency RTMS are used, and it is showing at least, okay, 2 weeks. Most of the studies is longer than 2 weeks. It also depends on type of studies and frequency of RTMS. High RTMS, you need lesser type of sessions. At least 2 weeks are seen in most of the patients. How long until the RTMS treatment starts working? So it is also variable. In old patients, it will take more time. In younger patients, they are having more chance of neuroplasticity, so it will show early. Okay, even in 1 week is sufficient for them to show the efficacy of the treatment. And mode of stimulation, high frequency, more efficient than the low frequency. Okay, so that's all about the general mechanism of the RTMS. Now I am moving to my study, what I did and what is my report regarding the RTMS. So I have selected not the depressive patient, but a specific chronic pain patients. They are fibromyalgia patients, and only female patients were selected in my study, as fibromyalgia is more common, 7 to 10 times more common in female than the male. So I selected fibromyalgia patients to show the efficacy, therapeutic efficacy of the low frequency RTMS. So fibromyalgia is characterized primarily by widespread pain and more than 3 months. And they are having, when we are giving slight pinch, which is not painful for the normal patient, but when we are giving touch or pressure, they will show, they will feel very painful. Okay, so that point where there is excess pain, that is called trigger points or tender points. So they are having out of 18, there must be 11 tender points as the old criteria for the selection of fibromyalgia. They are having pain in all but 4 body quadrants. They are having more fatigue, tiredness, muscle stiffness, sleep disturbances, depression, anxiety, and old patients, not always, but some patients reported cognition problem also. Poor, and due to these all problems, they are having poor quality of life. Another thing is they are more depressed because they are mostly drug resistant. There is no proper treatment except only the symptomatic relief by the analgesics, by antidepressants, they are given. Again, they quit the drug, again, problem will be same. So typically, the fibromyalgia patients were selected with the middle age, and this is showing what is the problem into the fibromyalgia patients. So it has been shown most of the lab tests in fibromyalgia patients. So everything is normal, blood as well as different hormones are under the normal range, but they are having chronic pain, fatigue, and there is very difficulty to do daily life activities also. So Stout et al. Studied diffuse noxious inhibitory control system as to control the pain in our body. There are different type of inhibitory and excitatory. So inhibitory systems like opioidergic systems, they help to inhibit the pain. So that inhibition has been lacking as shown by the Stout et al. Okay, in 2003, 2011. Okay, so vibrotactile stimulation is normal, no difference in control and fibromyalgia patient, but pain modulation, when they are checked like diffuse noxious inhibitory control, that is abnormal in them. Julian, it is showing increased pain intensity in fibromyalgia patients. Okay, and it is showing the same type of result that they are having endogenous pain control system is having problem there. Another, this study showing increased pain intensity means central sensitivity. Most of the studies show in fibromyalgia, sensitivity of the pain has been enhanced. Okay, so excitability is increased, so that is causing more pain than a normal. We use the term allodynia also. So how they are managed? Non-steroidal anti-inflammatory drugs are generally given, antidepressants, GABA agonist, physiotherapy. Okay, but they are only causing symptomatic relief. And this now they are given repeated transcranial magnetic stimulation, okay, as a clinical trial. So other studies like Samson is showing here, they have given 1 hertz, that is low frequency, for 5 days a week for 20 days on the right dorsolateral prefrontal cortex. And they assess the pain by visual pain scale, okay, and different type of questionnaires. And they show that there is significant improvement in the pain there in these patients. Mala is showing high frequency stimulation, okay, for 3 months. So they are also showing increased cortical excitability as well as improvement in pain. So both low frequency as well as high frequency, and this is showing low and high both there. So they are showing the big depression inventory. So depression, visual analog scale, everything is subsiding, okay, in both. So low frequency is working more in comparison to the high frequency. So in my study, as we selected the fibromyalgia patients, so we studied pain sensitivity, pain modulation, as well as before the study and after the treatment for 4 weeks. Okay, 5 days a week means total session given is 20. Okay, 20 session. How fibromyalgia are selected? As I told, fibromyalgia having 11 out of 18 tender points. Pain is more than 3 months, and only females are selected. At they are not having menopause, okay, normal menstrual cycle, and exclusion criteria is pregnancy, lactation, medication, different type of anti-inflammatory, chronic diseases like diabetes, FM, arthritis, or the patients having contraindicated to the transcranial magnetic stimulation like metallic implantation, pacemaker, seizure, brain damage. Okay, that type of patients are excluded from the study. So total patients selected are like 118. Out of that, SAM group, that is SAM stimulation given, RTMS group 59. Okay, and these parameters, normal hormones, etc., is also compared with the female normal controls. Okay, what are the parameters taken for the study? Is like pain modulation status. Its own endogenous pain system is measured by the diffuse noxious inhibitory control system, opioid status by Sucru challenge test. I'm not going to discuss these things. Only very easy, and that is clinically done. That is by the visual analog scale, McGill pain questionnaire, anxiety and depression by the state trait, anxiety inventory questionnaire, and BDI questionnaire. So, and quality of life by the WHA standard questionnaire there. So this is showing the study plan as the TMS is given for the 4 weeks. Okay, 2 weeks gap, 2 days in a week gap, continuously 5 days, then 2 days gap, generally Saturday, Sunday, then again another week. So after all the parameters like how much pain subsided, it is reported at the end of the every week. Then it is followed up for the 6 months. This is showing the age of the patients, control and FM. There is no significant difference, height and weight. Okay, so now I'm coming directly to the summary. So after the treatment for the 10 weeks, it is showing that significant decrease in the pain sensitivity. That's why pain is subsided in patients significantly. And the frequency used was 0.5 Hertz. And it is used for 4 weeks. So that improved chronic pain. And when pain subsided, they are having better quality of life, depression, anxiety, all parameters gone improved. And when it is followed for 6 months, there is sustained effect was seen in the 6-month follow-up also by both all the subjective and objective methods. And they are working abnormal endogenous opioid status in FM was rectified. Means we done diffuse noxious inhibitory control system as well as sucrose test. So it is showing that it is working under by causing the neuronal changes, neuronal plasticity, which will improve the pain and associated symptoms itself directly. That is the our study is also okay how this RTMS is working. It has been shown in normal persons also that causes increase the bilateral pain tolerance. Okay, cold pain tolerance in normal patients as done by the Guerrero. Samson also showing in the FM patient, same type of parameter they use, and there is significant improvement. So from our study as well as discussion showing RTMS attenuates pain-induced neuronal activity in all pain processing brain areas, including ACC, thalamus, midbrain, medulla. Okay, and there are some studies, they have taken the different type of opioids and they measured, okay, before treatment, after treatment. And it is showing that it is improving the opioidergic system to improve the pain. So almost we are at the end. FM is fibromyalgia is a chronic pain syndrome exhibiting all components like sensory, affective, motivational, evaluative. These are done by the questionnaires. And chronic pain in FM is of central origin, as it is showing the improvement by the RTMS. And RTMS is having long effect. Okay, so thank you all the participants for your patience hearing. This is the photograph as I am right now working in the Nepalganj Medical College, and it is inside the national park called Bardia National Park. And this photo is taken by some Bollywood actor long before, I think, 10, 15 years back. They saw all the tiger together. So thanks. And this is the college, Nepalganj Medical College Hospital. Thanks. Any question? You are welcome. Thank you.
Thank you so much, Professor Ansari, for a very informative talk. So I have two questions. One, does this treatment has to be given continuously to the patients, means intermittently, to relieve the pain, or we can give only for a short duration, then it can be stopped and can be, there is no need of giving it again?
Thank you, ma'am, for a very pertinent question. Actually, I worked there in All India Institute of Medical Sciences. As we know, patients coming there after trying all the places, when they don't have improvement, they are coming. And these patients, they are not getting any relief by taking the continuous medicines continuously. So we have a standard protocol. We are giving every day almost 1 hour for 4 days. So some patients, so immediate improvement, some it took 2 weeks, but according to protocol they are taking. And most of the patients, they completed 4 weeks treatment. Okay, and we were following up them either by the telephonic conversation, or they are coming regularly. So they saw improvement, and few patients after 6 months, they themselves requested that I have some problem again. So they took another session also. So according to need, we can modify.
So there's one more question, like, can this treatment be given to the autistic patients? Because there's no treatment as such. Can it be beneficial for them?
It is under clinical trial there. I saw, yes. Like eating disorder, this autism, Parkinson disease, Alzheimer disease, all this is the new technique. So everywhere it is now people trying to apply.
Thank you. Thank you, sir. Thank you once again, Professor Ansari. It was really a very nice talk. Now over to Dr. Anita George.
Thank you, Dr. Agarwal. And also thank you, Professor Ansari, for that very informative presentation, and for so readily answering our questions. So we have now come to the end of our 2-day symposium on recent trends in brain research. And I'm sure you will all agree that we had the privilege of listening to a series of exciting and inspiring presentations. The symposium has performed a valuable function in that it has brought together distinguished experts on many different aspects of brain research, and we have gained valuable information on neuromodulation and neurological disorders, techniques in brain research, neuronal plasticity and environment, and neuronal injury and repair. And so first and foremost, on behalf of the organizing committee of the symposium, I would like to express my deep appreciation and thanks to the speakers for their immense contribution over the last 2 days. We are also grateful to Professor Pixie Che and the International Brain Research Organization, Asia Pacific Regional Committee, for sponsoring the symposium, and also for giving us an insight into the activities of IBRO. I'm confident that many of our young participants will receive the inspiration and opportunities to embark upon research in the field of neuroscience as a result of this symposium. I would also like to extend our grateful thanks to Professor Suman Jain, Vice President of the Indian Academy of Neurosciences. As our scientific advisor, Professor Jain has been guiding us through each stage of preparation for this symposium, and we are really thankful to her. Our profound gratitude to our director, Professor Geeta Trilok Kumar, for her encouragement and support. Thank you, ma'am. And my thanks also to the organizing team and the support staff, Dr. Manjula Suri, Dr. Sunita Agarwal, Dr. Vandana Sabharwal, Dr. Bhupinder Kumar, Mr. Imran Pare, Mr. Rakesh Ranjan, and Tinsy Philip, whose combined efforts have ensured an interesting and meaningful experience through this symposium. Finally, I thank all the participants. I hope you've enjoyed your presence at this symposium, and it has provided you with new insights on recent advances in neuroscience research, along with the application of classical or novel methodologies in understanding brain functions in health and disease, and in developing new therapies. I would like to officially close this symposium, and I wish all participants the best. Thank you again.
All the participants can open their video so that we can have a snapshot, the final picture of the symposium. Those who can, please. Everybody, please.
Thank you, Dr. Thank you, Ajay. It was a pleasure listening to all of you. And thank you for sparing your time, especially with time difference, Ajay. Thanks a lot. And Professor Khindi, thanks for changing your schedule at last moment. Thanks a lot. Thank you, Professor Kiran Mai, for attending both the days. We are really honored by that.
Thank you, everyone.
Thank you.
Okay. I'm ending the meeting now. Thanks, everyone. Take care.